what we call it, manuscript. And definitely because it's a manuscript and it's a scientific document, the most important thing is if you want to publish your manuscript, we have a standard format that we need to follow, which is the logical structure of any scientific paper. And normally you write in a logical way so that at least we the readers will be able to follow what you are trying to communicate, so that it will be easier for some of us to understand exactly what you are trying to say. Now, because of this, and you are going to follow some logical order, your paper will only be accepted if only it has been reviewed by expert or maybe specialists to give some credit to it or refer it. Then we can say it has become a scholarly paper. And of course, not only a scholarly paper, it must have some scientific merit. Right, that being the case, we should try as much as possible as we communicate our manuscripts. We must have some clear expression in the way we write, but the expression should not be informal. It should definitely be formal, so that the key context of your, what you call it, your manuscript is there without missing anything. And of course, for the intended readers to also appreciate what you are trying to communicate. And especially this is very important for those who are the non-native, those who are not native speakers. Certain expressions might be very confusing. Now, if that's the case, we all know if you are writing a manuscript, these are the basic sections that we are all familiar with. So we should be able to go through all these things by the end of this talk. Now, one thing that we should look for is how comprehensive your manuscript is. I talked about legibility, readability some time ago. Perhaps I may focus on what we call how comprehensive it is. Now, when we say how comprehensive, the scientific norm within which you obtain your data and then try to scribble your manuscripts must have some of these elements. And we are not sure whether your work was hinged on a law, or you are talking about theories, or what about principles, mechanisms, trends and concepts, assumptions, limitations, significance of the work. Are you providing some good explanations about your work? And do you have evidence? Here we are talking about evidence and not proof as to back your claims. So if that's the case, we should also apply example. I'm just recapitulating what we have been doing. You realize that in every aspect of your manuscript writing will entail some sort of thinking, whether we like it or not. Every segment of writing will entail thinking. And here we are talking about abstract thinking, analytical. Later on, we'll see creative, concrete, conceptual. Those of you who have been attending my class, this is not new, but for the sake of the new ones, critical, convergent, divergent, holistic, and sequential. I have already talked about this in 2 of my lectures. Those of you who have messaged, maybe you may want to review some of my talk. Now, once you have all these, the logical framework, and then you have the reasoning capacity, now we need to zoom them down to what we call inferences. And when you are making inferences, I'm not a logician, but at least these 2 are the basic ones that we normally deploy in our work. That will be inductive reasoning and then deductive reasoning. Inductive is from general to specific, and from specific to general will be your deductive. So as we all know, we are all familiar with this. When you are talking about the inductive reasoning, here you are making some specific observation and then you draw a conclusion. If you don't mind, let's say we here in Malaysia, let's say there is this perception that, excuse me to say, which is very unfortunate, every black is a potential suspect. You get what I'm trying to say? So this is inductive. Maybe 1 or 2 have misbehaved and we have all been what? Blackmailed. So this is more to inductive. But if we are going into it and we are really going to take a good sample and we follow theory, we will know that actually in every society there are bad nuts. Therefore, the principle is there, the theory is there. So when we talk about that with hard facts, or we have some empirical data, then you are talking about deductive reasoning. That's why when you are talking about deductive reasoning, Normally we move from theory. There is a theory that you are hovering around, and then you create your hypothesis, your observation, and you confirm. But the opposite is true for what we call inductive reasoning. So inductive reasoning normally opens itself for what? Falsification, right? So once we get this, let's now go straight to the title of our, what we call it, manuscript. The title is very important. We shouldn't take the title for granted because it creates an impression. You know, first impression is very, very important. And that is exactly what the reviewers will see. So if it's something of less quality or has no value, then you are going to miss the boat. Now, it must be catchy in a way that it must interest the readers and even the editors and the reviewers. However, much as you want to catch the interest and then you want to create an impression, we wouldn't want to have too many words. I mean, there is no any, what we call it, a conventional number of words, but generally, if it's less than 15 or around 15, should be fine, right? Now, when we say fewer words, all that we want is the fewer words that you are using to describe the content of your manuscript must hinge on the issue or the gap that you are filling. We don't want any fancy or any extra words. And then it must also have, I mean, give us an idea as to how innovative you were to handle the gap that you are talking about. I'm not talking about general methodology. I'm talking about your innovativeness that led into the gap that you found and how unique your work is going to be and how you are going to achieve your objectives. Now, it should be able to also give us an idea, at least about the outcome, what to expect from your work, assuming we did not read any part of your work and then we are just reading the title. It must give us that kind of hint. So that is what we mean here. Now, as we are all aware, a good title, if once we read the title, it must be descriptive and it should help us to predict the content, at least, if not 90%, maybe at least 60, 70% before we zoom down to the what? The abstract, introduction, objective, and to the other end of the paper. Now, much as we want it to be very concise, specific, we don't want it to be too technical also, such that only experts or specialists will understand that your topic. I think we need to open a little bit so that at least what you call transboundary, people from different subject areas could also benefit from your work. Now, that being the case, this is an example. If you look at this example, we say the use of agro waste. I'm sorry, my background is in agriculture, so I may be a little bit biased. So, and then I may compel you today to learn a little bit agriculture. So when we say the use of agro waste, industrial agro waste to improve phosphorus availability and use of tropical peat soils, I think this is a very simple whatever. All that you are trying to say is you are using waste, agricultural waste, and then you want to improve something called phosphorus, and then improve it on what? Acidic soils. Fine. This is non-technical. Now, but what it tells us here is you can see clearly that we know, we can clearly understand the outcome. You are going to use a waste to do what? Maybe to improve nutrients so that we can what? The soil can be what? Efficient or healthy. Very straightforward. Sometimes also we have a title that could be more effective, can tell us the end of your story. So you see, students who smoke get lower grades. We can already expect what is going to happen. So these are the kind of titles that could be very, very valuable when you are talking about it. Now, the non-technical aspect that I was trying to say is this one. If you read this title, you will see that the first part you will understand by something like Fourier transfer infrared spectroscopy. It is too technical. So that if you are not in that field, you might not be able to understand it, and you have to go and read what is this Fourier transfer infrared spectroscopy. So in that case, it becomes a little bit technical. So if you have certain words that could describe this in a layman's understanding, that would be very useful. Now, much as we don't want you to sound technical and we want you to be as sweet as possible, it's also essential that you don't provide general, what we call it, the titles that are too general. Remember, titles that are too general are normally for what we call books, textbooks or whatever. So when you see something like this, sustainable management of waste, You see, sustainable management, sustainability is very, very subjective and it's a very broad area. And you are talking about waste. What are the wastes that you are talking about? So this would not be a good title for any serious manuscript for any recognized journal. Now, you also have to write the title in a way that it set the tone, just like when you go for international conferences or you go for intellectual fora, you see that we have keynote speakers. The role of a keynote speaker is to set the tone or the slant for the conference. So it's the same thing. If you are also writing a manuscript, you need to set the tone so that we can follow the logical, what we call a sequence, and then what to expect and what to imbibe. Now, this will help in a way, especially when you have very good keywords. Very good keywords, because if you have the good keywords in the title, then when we do the search, at the end of the day, your paper must be what? Read, and it must be retrieved also. So if it can be read, then it means that you have very good keywords. Now, there are schools of thought. We have a schools of thought about title. Now, whatever the case may be, what you need to do is follow the format of the journal that you are going to publish, you intend to publish your manuscript or your paper. So here, these are the thoughts. There are 4 of them. 1, certain journals or one school of thought will say that all words in your titles should be capitalized. All words, okay, should be capitalized. If a journal follows this kind of format, then you have to follow. Some journals, they say only the first word should be capitalized. If that is the case, you follow. Some will say nothing at all. You have to follow if that is what you want. Others will say only the first word and the nouns are the ones that should be capitalized. So here there is no argument here. So long as you follow the format of a particular journal, there shouldn't be any argument. But what we are trying to say, don't be a little bit fixed-minded in a way that when you see something that you are not familiar, you think that somebody is not doing the right thing. So that is how it is. Now, so example, this is it. The one that I just said, you see the first one, every word has been capitalized. I mean, for not every word, but the first a letter for every word. When you follow the second one, only the first one. The third one, nothing at all. The last one, almost every word had been what? Capitalized. So all depends on where you are publishing. Now, as much as you are trying to write a good title, it's very important that you are aware of what we call low impact words. Low impact words are something like a study of, investigation of, impact of. These are all redundant and they are extra words that are not very useful. So if possible, rephrase or reword your title in a way to avoid this kind of whatever. Can you imagine you are writing a review and you say a review of? Some of us, when we started and we were not very sure of what we were doing, we also used to, write in this way. But now that you are aware or you have been told or you have been, brought into this right perspective, it's good that we should avoid these kind of things. Now, maybe this is a little bit of science. I'm sorry, because I'm from a science stream. I may be a little bit biased. Here, if you are talking about scientific names or species, it's very important that you specify the scientific name, even though you have given the common name or the species common name. So here, example, if you look at this title, the issue is on maize. So here, the scientific name will be Z maize. And then the Z maize, you see that it's in italic. The first word, you must capitalize the first letter. And then the second word should be a small letter. L is the one who, Linus, the one who named maize as Z maize. So in the science stream, this is very essential, right? Now, if you conduct a research and it's a country base, then it's very important that you mention the name of the country if the research is limited to a particular country. Now here, remember, if you are talking about the country, then you should also make sure that even if it's within a town or a state or a region, and you want to highlight the town or the state of the region, you need to make sure that your sample or your research covers significant part of that area. If it's only a small part and it doesn't represent the town or the state or the region, then it cannot even represent the country. So we need to be careful and be aware of this. Sometimes a research can be regional, like maybe West Africa, you are talking about Southeast Asia, maybe the Middle East. So here, definitely we are talking about cutting across different countries. So in this case, we may want to know if it's a region, you specify the region. If it's a continental, you specify the continent. So here, this is why in this example, you see tropical. That means that the research was conducted under tropical conditions. So here it could be a little bit regional or zonal. Then that is how we look at it. Now, we should also understand that abstracting and indexing, you are talking about Scopus, you are talking about Science Citation Index, Google Scholar, and all sorts of things. So here, you will have to have very good, what we call a title for, indexes to index your work so that it can be retrieved and then get to the target audience. So I think that is the keynote that I would like to share here. Now, for example, if we were to go through this exercise, a quick one, waste management in different West African countries. So if I were to review this, or if you were to review, if we have time, 1 or 2 minutes, we should be able to comment on this. But I think what I'm going to cover today is very extensive. So I will try to share and then answer at the same time. In the first place, if you look at the title, different is not supposed to be in the title. Because West Africa, we know West African countries are already different. So the word different should not be in the title. Number two, if we read the paper and we find out that your research was only limited to, for example, 2 countries. Let's say you are talking about Ghana and Nigeria. You cannot use the word West African countries. You need to specify the names because you are dealing with only Ghana and Nigeria out of 16 countries. So there is no need for you to use the word West Africa. However, if you are covering a number of countries within West Africa and you draw a sample to represent West Africa, let's say out of 16, maybe 7 could be a good representative of West African countries, then you are qualified to use the word West African countries. And then waste management, what type of waste? There are different types of waste. We have agro waste, we have agro industrial waste, and is it a plant waste? Is it an animal waste? So all these things will have to be specified if you really want to write a good title. So this is an example where the title looks comprehensive. And the only thing about this title, if you are very, very familiar, this is why writing a title, writing what we call a keywords, you have to be very, very knowledgeable in the area. Like me, if I look at here, now you are saying amending phosphate fertilizers. There are different types of phosphorus fertilizers. So we will have to know how many of them. If 1 or 2, just like the example I gave on West Africa, you can apply the same concept here. It looks nice, but that phosphate fertilizer is incomplete, right? Now, if that is the title, let us relate the title to keywords. And you see a number of us, normally we take keywords for granted. If I were to quiz you, we will see who is really doing the right thing. So keywords are not for fun, or they are not just to meet the standard. They have their own place. Like I've been saying in most of my talks, sometimes keywords will help you to, let's say, like a journal that you are less interested in that journal. But you Google or you go through the databases and you just type very good keywords, and then let's say the keyword of the paper matches with yours, you could have retrieved it. Let's say I'm in agriculture, and then maybe I was looking for something on microbes or maybe on soil health or whatever, but I use the right keyword and I'm able to retrieve, let's say, a good paper from the medical journal. You see, this is the role of keywords. So it's not only in your area, sometimes where you have less interest, but you'll be able to draw some information from there. The other aspect of keyword is also sometimes it's the same, you are in the same field, you have been reading such journals, many times, but because of poor title. So keywords are actually backup for a title. So irrelevant title, you might have missed it, even though you read a lot of papers from a particular journal that such a paper was published. The only way that can help will be through what? Keywords. This is why when you are writing a paper, even if you miss a little bit in the title, make sure that your keywords will save you so that you get good readership. Right. Now, let me test you and see if I were to give you this title, and then maybe I requested that you kindly help me to write the keywords from this title. Let's see who can help. I mean, you don't need to be an expert in this area. I'm forcing you to move from your comfort zone and come here. And then just tell me, what are the keywords that you think we can get from this title? The moderator, I leave it just for a few minutes. Anyone who wants to say something? Anyone?
Okay, if you want to say something,
yes, anyone, just tell me what will be the keywords. All that this research is about is the same thing. They want to use a sustainable way of managing, let's say, phosphorus. We all know phosphorus, but you are using organic amendment from agricultural waste. So even you don't understand the concept or whatever, from the title, what do you give?
Please raise your hand if you want to answer for me to unmute.
Just contribute.
Or you can type in the chat box. I read.
Just few seconds. I have a lot to cover.
Okay, from the chat box, we have agricultural waste.
Okay, somebody say agricultural waste. Let us keep it agricultural waste.
Okay. Another says phosphorus.
Okay.
Phosphorus fixation.
Other says
sustainable development.
Okay, phosphorus fixation, and then what else?
Sustainable development.
Okay, sustainable development.
Then we have a phosphorus, organic, agricultural
waste.
Phosphorus,
organic, agricultural waste. All right. Okay, thank you.
Another one, organic amendment.
Okay, amendment.
And then the last one, management, phosphorus fixation, and agricultural waste.
Thank you very much for your contribution. Now, all of you, keep your answers and see once we finish whether what you said are within what we want when we say keywords or not. So let us start now. Now, when you are writing keywords, normally it must be derived from the subject matter. So first, for you to write a very good, to write good keywords, you must have control of the subject, the paper, the manuscript that you are writing, the context. You need to understand that. In relation to what? Now here, if I were to write the keywords, the key thing about keywords is never repeat any keyword that is found in the title. It should never serve as a keyword. Never. We don't, even though we do, you hear people writing just like what people are saying, all that you are telling me the keywords are from the title. No, you don't have to even pick a single word from the title. That is what should give you keywords. And this is a backup to the title. If you repeat the same words in the title as your keywords, then the examples that I gave you, your paper will be missed, and it might not be read very well, and your H index will be poor. So here, if I were to write the keyword now, I will be looking rather for the agricultural waste. You get it? I'll come down. Then the name. Then the organic amendment, I would not want to use organic amendment, but I will look for the state of the organic amendment. Is it liquid? Is it solid? Something like that. And then next one, I will also look at soil type. You get what I'm saying? Is it an organic soil? Is it an inorganic soil? Something like that. This is why you need to have the knowledge. Then the third part also, I will look at the method that was used to produce the product. So these are the areas that you should look at in a way that you don't repeat the words in the title. You will see later. Now let's see. First, if let's say, example, the waste are very, very exclusive to a particular region, or let's say a zone. Let's say like you take pineapple. Pineapple is a tropical fruit. So here, if it's in a particular region, then you are qualified to use an example, tropical, subtropical. Let's say Ghana, we have something called Guinea fowls. They are very exclusive in certain parts of that region. So you can use the word region, I mean, these kind of regional names. And then if West Africa, whatever. So here, you need to understand the subject before you start going for your keyword. This is one example. Now, another example is just now I was telling you, we have to know, you say from waste. So instead of us to use waste that you have in the title, we would rather mention the names of the waste. So if the waste, you are talking about pineapple leaf should be your keyword, rice straw, something like that. These are all waste. So you don't have to repeat the word waste in the title. Now here, some of you said phosphorus. Yes, but what type of phosphorus? Now we have something called triple superphosphate. You have rock phosphate. You see now why I'm telling you, you need to have a control over the subject matter. Now here, waste. If you are talking about organic amendment, how do you produce it? What is the material or the machine, the equipment? Normally we use composting bin. Now, if you want to, we say organic amendment. What type of organic amendment? It can be compost, it can be biochar, it can be organic fertilizer. Now, where are you going to use the organic fertilizer or the amendment? On acid soil or alkaline soil or basic soils? So these are the words that you should use, and you will see that by the time I finish, none of the words in the title will appear in my keywords. Next. The next thing also, you need to consider the experimental techniques. So here, what are the techniques? If you are producing organic amendment, normally we do composting, we do bioassay, we do pyrolysis. You see, so here you use those techniques instead of the words in the title. Then we can also, in what context of agriculture are you going to, use that kind of organic amendment, or you are targeting? So here we can say, if it's organic amendment, you can say organic farming. You can say biological farming, soil fertility improvement, improved crop yield. You see, so these will become your what? Your keywords. Now also, sometimes the consequences of your what? Your innovation or the problem that you are solving. So here, eutrophication, anybody who is in this field will tell you when you use too much of fertilizers like nitrogen and phosphorus, you will pollute the environment. That's why you see certain water will become very greenish, algae. So here, if yours is organic, you definitely will talk about environmental issues, and that is what we call eutrophication. Now, if you're here, you are talking about acid soils, then we will have to talk about something called aluminium iron. If it's an acid soil, you need to neutralize it by what means? Liming. Now you are coming out with an amendment. What will be the economic viability? So here you can use the word economic viability. You see, I'm in crop science or soil, but I'm still using the keyword for economic viability. So somebody who is working on resource economics will be able to retrieve my paper. Value creation, because you are adding value to the waste. So these are some of the keywords that you have to use. Now let's go back to the exercise. From the examples that I gave you, can you really see from here whether I repeated any word that is found in this title? If you really understood what I'm saying, I never use any word that is in this title. So that is the way you should write your keyword. So from today forward, keywords, never extract words from your title. Create a situation that you will have a safety net for your paper to be retrieved, but it must be relevant to the subject matter of your paper, of your manuscript. This is why I'm trying to say here, note, avoid using of phrases in titles as keywords. So from today forward, you may want to avoid that. Now, from the keyword title, let's go to the abstract. Now, when we talk about abstract, remember, technically, abstract is just a one paragraph. It's a single paragraph type of what? Write-up. And normally, generally, the word should never be more than 500 words. In fact, in certain journals, they will limit you to 120 words. Some will say 300. Some will say maybe 250 or 200. but generally 500 is considered too much. So again, it depends on the general requirement. By 500 words for an abstract, actually it's not a good abstract. Abstract generally will summarize the key content of a manuscript. So you are just telling them the gist of whatever so that we will be able to read more. So that is all that an abstract is about. This is the reason why when you write an abstract, it must be short, concise, but very informative. And this is another reason why, remember, I've been trying to, adduce you to, to let you understand that whenever you are writing, you must have the flair of writing, good choice of words. The right words, use them at the right time and try to compress your words as far as you can. Otherwise, it will be very, very difficult for you to write an abstract. Now, what are the components of abstract? One, we must know why the research was conducted. It's very, very important. And how it was conducted. I'm giving asterisks because later you will see that depending on the format of the journal, certain aspects may be missing. That does not mean that your abstract is not correct. Now, abstract must also have key results with brief interpretation, very important results with some a bit of discussion. Now you need to conclude your abstract, and if possible, you provide implications. So this is a general format. But now let's look at this. Again, just like title, there are schools of thought about abstracts. Now, when you are writing an abstract, it depends on the format of a particular journal that you are targeting. Some journals, they don't require the background of the problem in the abstract because they feel that it will be in the introduction part. So now, if that is what you are going to follow, then you follow. Other parts of the abstract that I just mentioned will remain. Some also, they want you to go straight to the objectives, straight to the objectives. They don't even want to hear the background. So if that's the case, then you need to follow. Other parts will remain. Some also, they are not interested in your methodology. So if that is what they want, and they want the other parts of the abstract, follow. These are some of the things, so that you don't go and see an abstract and you start criticizing without knowing the format. So these are all acceptable, and they depend on the kind of journal you are writing to. Some also, they don't need interpretation, they only need the key results. Of course, with the other parts will remain of the abstract. Now, some also may not need implications of your findings. They might not need the implication. So if that's what they want, then you go ahead. But some journals will want every part of an abstract, a very standard type of writing an abstract. So I'm going to run you through a standard type of abstract, but wherever you go and then you have other requirements, then you may want to follow. Now, a well-written abstract, generally, the problem that was studied, you just write it in 1 or 2 sentences, and you must focus on why the study was conducted or why it will be carried out. So here, only 2 sentences should be enough to give you the problem. You get what I'm saying? Because it's an abstract. You have, let's say, 200 words. Now, number 2, within the abstract, we must have a purpose or the objective. Okay, also 1 or 2 sentences, but you should focus on what the study was and how it differed from others. That's all that we want to know, because you don't want to repeat anybody's work. So here is very important that this simple thing must be there. And the third one, if you have the methodology part, also 1 or 2 sentences. When we say methodology, your creativity, we are talking about your creativity that makes your work special. So it must summarize the important or the key methods. That is what I'm saying, the key methods that you are using to solve the problem or to come through the study. So here, these are the things that we look for. Now, number 4, if you are following the standard format, we are only interested in major results, only important results. and not all results, because some may be just supporting results. We want the heart of the word of the manuscript. Report those only. The rest we will go to the full text and we will find them there. Now, results interpretation does not mean you create a whole page for discussion. This is not the place. All that we want is the key important things, and here just for you to add value to the results. That's all that we need here. Right. Now, number 6, if possible, to create significance of your work, it will be good if you have implications of your findings that has been interpreted, the key ones. So this should tell us what we should care about your paper, especially in terms of conclusions, recommendations, or future directions. That will make us care about your work. So these are the components of any standard abstract. The rest I have told you, some journals may not want this part, or want, for some reasons, you don't have to follow. But whether you like it or not, this is an example of a standard format. Now, much as you are writing, also be aware, there are certain things that you should never include in abstract, tables and figures. I'm not sure about your field, but we in the sciences generally, unless maybe you are talking about mathematics or computer science or something like this, generally tables and figures are not included in abstracts. And if possible, also avoid excessive use of acronyms. In fact, some abbreviations and acronyms, unless they are very standardized, well known, it will be very, very important that you reduce. Sometimes you don't have a choice in your research. Yes, certain things may be repeated and they are going to be long. So you want to give some acronyms or abbreviation, that will be fine, but it shouldn't be excessive. Especially, for example, DNA, RNA. These things, we don't have a choice. You got what I'm saying? But what I'm trying to say is reduce the excessiveness. Now, also remember, you should never reference an abstract. There's no need for you to cite references in abstract. Unless your methodology is dealing with method. So it's very, very important that you need to be clear about this. Now, abstract also, remember, never write any scientific manuscript without an abstract. It's never done anywhere. A scientific manuscript will definitely have an abstract. That is the third point of call. It's very important. Now, also, if you are writing, make sure that the background of your abstract is not too long. Because once your abstract is too long, the background in particular, you might lose out in the important results. You might be constrained to even conclude. You might be constrained to give your procedure, your methodology. So don't give too much of background information. That's not the place. The place will be in your introduction. Now, sometimes also we write abstract with this kind of missing information. Unless you are following the journal format, at least an abstract should have a purpose. If we go by the conventional one, you must have a procedure, major results, what the results mean, and conclusion. At least these things should be there. Right. Also, if possible, what you can do is you need to have some proportion, background, problem, method, results, interpretation. You got what I'm saying? Lengthy, whatever here is not acceptable in anywhere. So it must be, that's why I say 1 or 2 sentences for each segment will be fine, or each section should be fine. Right. Now, that is why here a sentence or 2 for these sections should be able to save you. And here you need to have rich vocabulary. You must have a flair of writing so that you can compress your words in a way that you carry the meaning of your work. Now, this is the issue. If you don't follow these kind of guidelines, remember, editors have no time to go through a lengthy abstract or an abstract that is not concise and precise. So here, remember, we are having a challenge of information overload. Therefore, it will be very, very important that you stick to this, because some editors, they just read your abstract. Once it's off that is where you end, at the editorial office. In fact, that is where your paper will end. So it's very, very important that we should take care of this. It will never even go to the what? Reviewers, I mean decks for them to start reviewing your work. So it's very important. Now, let me give you some few examples and then we will see. Let's see, look at this. What do you think about this abstract? It has been published in a certain journal. I will never reveal anybody's paper here. So what do you think about this abstract?
You can type your thoughts in the chat box.
A quick one, anyway, just one person or two so that I can move on. What is our impression about the abstract? A quick one. Sorry, today you are learning agriculture. Most of you don't like agriculture, so I'm using a strategy to prop your interest. Okay, I just want yes or no. Is it a well-written abstract? For me, I think this is not a well-written abstract. A lot of information are missing here. And I don't believe that any reputable journal will accept this kind of abstract. That is why you need to publish in recognized journals. This doesn't augur well of you if this is a kind of abstract you read. I mean, you write.
Yes,
right.
All the comments in the chat box, I agreed with you, Prof.
Okay, excellent. That means that we are on the same bearing. But what about this abstract? This abstract.
Somebody says no, not okay.
Okay, somebody said no, not okay. So this is how we segment the abstract for you to see when we are reviewing or when you are on the editorial board. You see, now we are checking. This first part looks like, okay, fine, maybe the introduction aspect. Now here, the problem aspect is a little bit there. And then now you go here, we can see the purpose a little bit, but it is mixed with a little bit of what? Methodology, a study, a greenhouse or whatever. Now here, you see, there's dwell a lot on the results part. And here, we don't even have the methodology part, and we don't even have any significant interpretation of the findings because they waste a lot of time, maybe giving irrelevant information. And you see, so when you have this kind of abstract, it's definitely not a very good abstract. So that is why you need to have the segment in place so that when you write, you don't fall into the trap of this. And then this is the conclusion. You see, we don't have the interpretation, we don't have the implication. Now, the next example is this. Again, what do you think? Sorry, you don't need to be very much bothered about the content. Just scan through. It should tell you something. You just scan through. You don't have to understand it. Just look for where is the introduction, where is the objective, where's the methodology, where will be the result part, discussion, in whatever. If you look at this kind of abstract, it has three components. The first one, I'm not able to understand, even if it's an objective or it's a methodology. Until now, I cannot understand this. And I feel that that is not, once we are doubtful about what you are saying, straight your abstract will be rejected. Now you look at this from here, right away to results. We are not even sure of the objective, of the background or whatever, and the whole of this is giving us. And at the end of the day, there is not even a good conclusion. So these kind of abstract will never go through. Have you seen a lot of what abbreviations here and there? So many abbreviations.
Somebody made a comment. So many abbreviations.
Exactly. So I think that we are in the same line. Thank you very much. Now, this is another one. Then here, if you don't mind, I just give you the clue. These are the segments. In fact, this is what I do in my research methodology class. Abstract alone can take us one whole lecture. So I give them assignment, they will go and review, and they come, we discuss or whatever. But what I want to show you is the first part is talking about the background of the problem. And the second part is just talking, what you call the objectives. And the third part is telling you the results. You get it? So here, if the format of this journal is that they don't need the methodology part, then this paper already qualifies in terms of not writing the methodology. But if we find out that the methodology part is supposed to be there and not, then it has skipped the methodology part. And then here, you can see here the last part. This red part is the interpretation part, a very brief one. And then the last part here is the conclusion part. This is relatively better compared with the other ones that we are talking about. But again, you are having a number of acronyms here and there. And that is another flaw of this kind of paper. Now, when you look at here, all that I want to tell you is when you go back and read, you will see that the components are there, but there is no conclusion and we don't have implication of this abstract. So again, this will be a problem. Ladies and gentlemen, I think it will be very, very not good on our part for any reviewer to comment on your abstract at this level, to comment on your keywords, and to comment on your title. That is not a good impression at all. So that's another thing. Now here also, almost some of the items are around, but there is no implication part. So when you have time, you go through and then you might see what I'm trying to say. And this is one of the aspects that has really shocked me until today. You see here, they have the aim, but the aim is mixed with methodology, if you read carefully. And then here you have method together, which normally is not really a universal format. I don't know your field, but we in the sciences, looks strange for us. Now, then the most significant part of it is, look at this: conclusion, significance, and impact of study. And look at this. You read, it does not reflect all these. These are very serious components, but when you read here, they don't really answer this part very well. So here, this will be considered as a very poor abstract. Right. Now, this one, I consider it to be a good abstract, but it has so many acronyms. But when you read through, it has all the standard formats. Assuming that we are judging this paper based on standard format, then it has all the components. So we call this a good abstract. And if you read, you will see the logical flow. Right. Now, what about introduction? Now, when we go to the introduction section, remember the introduction section is an expansion of your abstract. So this is why we say that it provides complete information. Now, the abstract is not complete because you are reporting only important information. So here, once we get the complete information that we need, it will help us to understand the rest. So once you read the introduction, at least you will have a very good understanding of what you should expect in the subsequent sections. Therefore, what we can say is an introduction section should provide what? Background information. Now you have to go detail, not like the abstract, and you must include the rationale for the research so that the reader is persuaded. You are enticing the reader to read more, so that when you chronicle your facts in a very logical way, then you are within what we call the introduction part. And it must be very useful and interesting. You don't just write without any coherence, and that one is not acceptable at all. Now, remember, the background information should be related to the need for the research. That means that the gap that you found must be related. It must be hovering around that. Don't go outside the loop. It's like a centripetal force. You are going through the cycle. Right. Now, remember, in the introduction part, this is where you build the argument. You build the argument and present your hypothesis, your questions, and your aims, which will be your objectives. So this is where the argument will be there, and this is where you show your strength in the gap, why we should listen to you, why, and what do we care about you. If that's the case also, we must also understand sometimes you have observed something and you really want to go into that with a very, very keen interest. So here, you must also show your personal motivation behind the research. It's not just like somebody is compelling you to do the research. Nobody gave you the topic. You are the one who you think that you are interested, you want to be a specialist in that, you are curious, you want to go further. Therefore, we must see your personal motivation. But your personal motivation here does not mean you say I, or no. And through that also, if there are some theoretical basis or points that you are trying to adduce, it's very important that you go through. And then here, the introduction also will outline the subsequent area of the manuscript that we are going, because again, it will tell us the background. What was the problem? What has been done? What has not been done? What you want to do and why? So you see, you are giving us the direction. So the introduction part is really work on that. Now, it must summarize what? This is a summary. It must summarize the background on the subject in terms of the problem. And then it must address the problem itself. So background is there. You give the story, then you zoom in into the problem. Right. Once you do that also, you cannot be doing it in a vacuum or isolation. You must refer to previous research work so that we know that you are not in isolation, and to add on to what? Your credibility. So it's very, very important. Now, as you do that, you need to explain exactly what your manuscript will address, why and how. These are very, very critical things that we will always look for. Once they are not there, forget it. Now, let me take them one by one. When we say the problem, what we mean by you have to describe the problem that you are going to study. And here, it must include the background of the problem itself. Now, you must summarize relevant research that provide the context. We need to know the context. What is the story? Where's the direction? And the key terms and the concept that you are trying to build on, we must know them. They should help us to understand your work. Very, very important. Now, for instance, you look at this. If you take time and read this, you can see that we are giving the background a little bit, and then we are giving the key concept, the principle behind the work in terms of chemistry. So here, once we get this, we start to build on it until we move. Right. So if that's the case, note this. Introduction part or whatever, as you know here, must be wherever you are introducing your subject, it must be backed by a very credible literature, if possible. Your thinking and too much of story doesn't work well. So you need to put the context in a way that you reference giving very key literature that are relevant to that subject area. Now here, as you try to introduce the key literature, make sure that you don't just only introduce the authors. You have to tell us the work and the major findings and how they relate to your part and the gap that you are trying to fill. Don't just tell us about the authors and you stop there. It doesn't work well. This is an example. We don't quote like this. Latifa Eta use sago waste and sago wastewater and zeolite to minimize ammonia volatilization from urea. This is not a good quotation. Anybody can do this. So this is not what we mean. What we mean is, what are you getting from that paper? So here, if you are saying that ammonia volatilization has been minimized, what was it? How was it done? Or what's the reason? Then you say because the former material. So here, the red part is adding the scholarly aspect of the review. This is how you review and put yourself in the context and within the gap. So don't just describe, but you need to have some intellectual debate or intellectual addition. Right. Now here, not only just the problem. No, we must know the importance of the problem, the significance of the problem. So here, you must provide pertinent reviews that will explain the following. One, The findings that your work hopes to challenge should be there. You are challenging something, you are adding on to something, or you are extending something. It must be very glaring. Now, findings that your work hopes to also extend, if you are not challenging, sometimes you are extending, do so. If your research is about conflict resolution, the debate is ongoing, the debate is there, and it is not, the answer is not there yet, and you want to come in and say that you are going to answer the debate or the question, it must show and it must be convincing. Now, sometimes it could be that it's an area that is very new and it's a new ground, and people are going there, and you want to show that you can champion the area, then you have to let us understand that part as well. So these are some of the things. So 1, 2, 3, 4, at least you must fall with, you must show that you fall at least within one of them. Out of this, then actually you are not presenting a scholarly work that will be that will get into any recognized journal. Now, much as you try to also talk about proposed solution, you are proposing solutions to the situation. It's very, very important that you give us some brief description on your experiment in relation to what? Hypothesis, research questions, and if possible, summary of the methods that you are using, and what kind of procedures, how innovative you are, how unique you are, that you are going to bring in something that we never know. So the choice of method will be very important. And you see that when I get to the methodology section, you will see how I'm going to relate this part to the methodology section. Now, what are some of the common mistakes in writing introduction? These are some of the common mistakes. Sometimes you just fail to demonstrate to the editors or the reviewers that you are really familiar with the area and you know where the debate is and where the debate should go and what is lacking in the debate, the intellectual discourse. If you fail to give that kind of impression, then you are just like a lab technician who has, or maybe like just a enumerator who has just got some data and just want to publish anyhow. So this is why you must explain these things very, very carefully so that you discuss on the main players in that area and how you want to contribute. It's very important. Now, sometimes also we fail to demonstrate our critical review and the way we analyze the situation so that we can contribute to the field in a meaningful way. So it's not just like you are just another type of paper who is reinventing the wheel. No. So critical thinking comes in, analytical, then what makes you different? You need to demonstrate that. Now you need to also demonstrate that you have exhausted the literature in a way that, I'm not saying you should read millions of literature, but the little that you know, you are very sure that you have sufficient literature to ensure that whatever you are going to research into or you are publishing, worth publishing. Now, failure to use that will be another thing. So one trick is you have to use what we call verb tense in a way to convey your subtle meanings. It's very, very important. So sometimes misuse of words could blur the whole information. And last 3 weeks, I did take my time to talk about this. Now, sometimes also, in the presentation of your introduction, you are just unaware that you are making a lot of repetitions that are not necessary because there is no chronology in your argument. There is no kind of flair. There's no logical, flow in your work. So these are some of the things. And sometimes you just don't understand what is plagiarism. Copy, paste, and then just rewind area here and there. So we need to try as much as possible to avoid these kind of things. And also, sometimes it's very unfortunate that you think that the theory or the area that you are talking about relates to your area, but actually you are citing or you are working on theory that is out of context. That could be it could be a very sad situation. That itself tells us that you really don't understand what you are doing and you don't deserve your manuscript to be published, and the credibility is not there. Now, the other aspect will be that sometimes we include information that too much information that are not really relevant for the manuscript. You know what I'm saying? Too much of information that are not relevant. Some of it we think 5 or 6 pages will be good, but no. When we say too much of information, just look at this. a lot of irrelevant information here. So when you start to write like this, you are just wasting time and resources. Let us review this a little bit. Another exercise. Needless extra words. So who can help me? I want this to be written not more than 3 sentences. Not more than 3 sentences. Any taker? Well, in the first place, the citation is even wrong. How do you cite like this? There's nowhere we cite like this. So in the first place, we don't take you seriously. Even you don't know how to cite. This is one aspect. All these things that you are trying to say, this is all that you are trying to say. Only 2 sentences from all these. So this, when we say that extra information, this is what we mean. A lot of information, but they are all irrelevant. So this is what that you want to say. This is why you have to write succinctly and to put the key context. Otherwise, you are blurring the whole information and that's it. Now, another example is sometimes you provide insufficient information. The first part is too much. Now, the second part is insufficient. Why? Because you assume that whatever you are writing, people know, or maybe the editors know, the reviewers know, oh, this is very, it's a general knowledge. If it's a general knowledge, then why are you saying this? It's a research that needs to be embarked on. So here, it leads to you not being able to explain concepts because you are taking things for granted, things lightly. You are not providing sufficient background information, and you are not discussing previous research in a very, very important way. So this tells us that you don't have the control over the subject area, right? Now, note this. It's very important that you have to be very clear in your writing and do not make the manuscript so difficult. So not too much of information, no less information. Just keep to track and put yourself in the right perspective. How you do this, you need to have a control in your area. That's why you don't do research, you don't have a gap, then you say you have a gap, then that's where the problem will be. Now, sometimes also you lack clarity about the work. So here, when you are having a thorough introduction, very, very comprehensive, every aspect has been covered, but what you lack is you are not clarifying the issue. So there is no clarity in your work. Clarity in a way that you are not being blunt, you are not being straightforward to tell us exactly. Too many stories. You get what I'm saying? Maybe you have the fact, half a paragraph, only 1 line is important. So this is why sometimes you can be blunt and say that this paper will cover. This is the reason why, how. So these kind of words will put you in the context of your writing, right? Now again, just like title, when you are writing, let's say like your research, if possible, to make it very straightforward, the study compares, this study examined, in this study, in this present, because sometimes if you don't use the word in this present study or in this study, if you are not very, very, definite, because you might be quoting certain references or relating to other work. So then we will become a little bit confused. So once in a while, when you say this present study, it sounds awkward, but you are trying to be a little bit specific so that you dissociate yourself from whatever you had already quoted. So this is the place. And then sometimes also, if possible, too many listing and bulleting in your introduction part. I'm talking about manuscript, too much bulleting, too much listing. If possible, reduce it. If you can write in prose form is better than try to bullet in introduction. I think it doesn't read well. It's not wrong, but it doesn't read well, right? Now, sometimes also we have what you call confusing structure. That means poor organization of your thought. If you are writing, then you should remember this. If you are organizing your thought, remember there is a bigger picture or there is a big area, but your part is just a microscopic aspect, just a small part. You are not solving the world's problem. So that is how you should target your work. Unless it's a transboundary, it's a continental research, that's a different thing. Yours is just a small part. And then also you are forgetting the underlying logic of your work, which is the hypothesis. These are the guiding whatever, I mean, these are the guidelines that should help you so that you don't go astray. And then sometimes also the description of your study. What you want to do is not clear. So these are very important things that we should understand. And then here, you are not able to tell us what is special about your work. This is very, very important. Otherwise, if there's nothing special about your work, then you are just repeating what is known. So these are the issues. And when you are writing also, as we say, you have to be very specific. Please, in manuscript writing, it's not any kind of writing like a music type of story writing or whatever. It's a serious type of writing. So don't open your writing with amusing things. Be very factual and straightforward. And if possible, don't bring yourself into the picture like I found or whatever. Of course, some people also have reservation on, I mean, a passive kind of writing. But it is well accepted in the academia that once in a while you can use passive kind of whatever writing. Some say you must use active or whatever. But whatever you do, the key thing is avoid bringing yourself into the picture. Even if you want to say I, that's why we use we. We is we of what? Plural of respect is very important. Now let us pause for some brief exercise about the introduction before I go into the methodology. So example, if you have something like this, I mean, I will not ask you to read this because it's going to be quite long. If you have something like this, all the components that I just said, you should scrutinize one by one and see. So when you go back and you read the lecture notes, try to see whether you will spot them out. If you have an eagle eye and you are able to follow, these are the points that you should be able to deduce or to spot out. So you can see the background, and then you can see the problem and why the research, and then what will be the objective, how unique they were, what were the methodology, what did they find, how did they, what are they trying to say that they want to do, and how special are they, what is their hypothesis, what will be the research question, and what kind of research and what they want to achieve. If you take your time and read the introduction, all these things must be clear. This is another example where you can see, but this kind of example is not proportional. You can see that the argument part is a little bit long. No, I'm just giving a very simple one. So these are some of the things that I can share with you. Now, let's go to the materials and methods parts. Here, basically, we are all aware. We just want to know, if we were to repeat your work, are we going to reproduce your results? I'm not saying exactly, but can we get just similar results? That's all that research, this section is about. So here, this is why it's very important that you are very exact. You are very, in fact, if your research method, materials and methods section is flawed, then just forget it. So here, you need to be very exact. This is why you have to tell us the experiment that you conducted or the research you conducted, exactly how was it done? We will have to know, so that if we want to replicate it, we should be able to do so. You also have to tell us what are the materials. That's why we say materials and methods. What are the equipment? What are the instruments that you use? In terms of what? How they were used. How much was used? How often? Where? What? When? And why? These things must be very clear, because without you, we should be able to read and try to replicate what you are doing. So these things must be very clear to us. Example, the fact that we say it's a methodology and you need to tell us detail about your work does not again mean unnecessary things or irrelevant things. Just take your time and read this. This kind of writing should never be in any scholarly or recognized journal, because it's like a secondary school or primary school type of research experiment. If you have been in the secondary school before, Form 1 to Form 3, before you move to art stream or science stream or social science stream, we all took some basic sciences or whatever. This is secondary school type of writing. So if you write this, nobody takes you serious. Just read. Pepsi was poured into a graduated cylinder until the bottom, meniscus. All kind of things. So this is not the way. So this is all that you want to say. A 45 ml of Pepsi was added to each of the 55 samples. That's all. So when we say you write exactly and then very precise, this is what we mean, not the lengthy type of writing. Now, on ethical grounds and also to reduce error, it's very important that you specify if you dealt with animals, how did you deal with them, plants, humans? How did you handle them in a way that whatever results that you obtain could reflect the findings? It's very, very important. Example, if let's say you are trying to deal with human beings and you are talking about you want to deal with cancer and you are talking about try to come out with cancer therapy, and you selected some samples and already you don't have a way to know what kind of cancerous patients are you going to work on or whatever, then just forget it. And even how do you condition them before you establish or administer your treatments? So these are very, very important. Now here, it's also very important that you should let us know your site. It's very, very important. Site information is very important. It could be physical or biological features. And the exact location, if possible, provide some maps. I mean, especially those in the sciences. So this is what I mean. If you look at this example, all the three things that I have said have been already provided. So here, if I segment them, you will see the study was conducted. So you know where the study was conducted. It is telling you about the physical aspect in terms of temperature, rainfall, humidity, and then we know that. And it's telling you also the type of plant, the species. So here, this is just a background information so that when we want to trace, you see, you have been given the coordinates. So we can easily trace where you did the work. This is very important. And sometimes you can even go further and give us the map of the area or the map within which you are trying to work. These are very, very important things that could really, really enhance your work. Sometimes we might even need some satellite data to see really what is going on, assuming you want to know exactly individual plants or individual community, a sociological setup, how does the area look like, we will have to know this. Now, we should also look into information like the procedures, how credible they are during your data collection, the experimental design, the statistical methods, what will be your level of significance. I was reading someone's thesis and they are using 2 levels of significance, which is very strange. Alpha 0.01, alpha 0.05 for the same finding. How can it be? This tells us that you don't really understand your statistical information and you don't even know how to interpret it. So this is very important, ladies and gentlemen. And then here also, we want to know how precise your data is, whether it's your questionnaires, you have piloted them, you have ensured that all the flaws have been taken care of. If you are in the sciences, the machines that you are using, have you calibrated the machine? That is very fine. It's just for you to tell us that the machine is working, but it doesn't tell us the accuracy and the preciseness unless you use a reference sample, the sample whose value is known to correspond with your data. These are some of the things to tell us about the validity and the consistency of your work. Now, it's very important that when you are writing in scholarly or reputable journals, if the procedure that you are referring to, if it's a standard procedure, there is no need for you to give us details. Just give us the name of the procedure and what it does and give us the reference. It's enough. Some, they want to write a whole lot of thesis on this. It's not necessary. So a standard method, just reference it and that's it. Now, it's also important that you summarize your research design and the methodology. What do we mean here? What is the philosophical construct or the philosophical framework of your work? This is what we call the epistemology of your research. Knowledge about knowledge. The knowledge about your research is what is important to us. How do you get this? You can get it within the context of the literature. Now, also, you have to present the rationale for the methodological approach. So why are you taking this approach and from what is known? Why do you feel that what you are saying is the right way to go? And are you sure that when we follow you, we get credible data? And you can only argue your way out when you are using the literature or you are expecting in that area. Now, also, how do you justify your result? Your methodology or describe your methodology? You might need some information from the literature. It's very important. Now, once you try to do all these things, it's able to review the boundaries for you, and then you focus on your boundary. Very, very important. Now here, if possible, it's important that when you do your description, methodology is something that you have done it already. So it's very important that you present your information in the past tense. Of course, there are different ways to write it, like present continuous or whatever. That is fine, but you have already done it. You are reporting. So when you are reporting, it's very good that you report in past tense because it's already been done. So these basic things should not really, whatever, elude you. And then also here, it's important that you show your timelines, especially when you are dealing with very important topic that has some sort of relevance and it shouldn't be outdated within a certain period of time. Example, if you are in the IT, area, for you to produce an information within 3 years is becoming obsolete already. If you are in the mobile phone technology or communication sector, so these kind of things, you need to have some timelines and then get them done. Now, the next aspect will be ethical issues. You see, you need to really show wherever there are, let's say, confidentiality. If there is confidentiality, you need to show. You see, when you bring in your ethical issues, it makes us take you serious as a very credible kind of researcher who will not just do anything anyhow. So these are the things that I think I want to share with you. And remember, I read one thesis and I was very happy. This person was very critical about validity, reliability, and all sorts of things. That's very good because you are adding value to your work so that whatever you say, we can say that that is a fine work. Now, let's do some exercise. This is a short methodology. I just want you to read this and tell me your impression. I'll tell you later. Just read this. What do you think about this method?
Yes, you can type in the chat box.
It's really unfortunate this is not a normal class. It could have been quite interesting. There are a lot of things that we could pull out from all these examples. All right. All that I want you to see is whenever you see that a method is being described in detail but very concise or whatever, it tells you this method is not a standard method. It is not well known across the world or in the intelligentsia or in the academia. So there is, if you are writing this way, it tells us, because you are giving every information, that tells us that this is not a standard method. And sometimes you have a hard time for people to accept unless you argue your way out. So this is an example where when the method is not a standard method, you need to describe and then make sure that it is well described. Now, that is what I'm trying to say here. Number two, what do you think about this? This is more to economics. So those of you with economic background, I think you can help us. Now I'm moving away from the ordinary kind of whatever, but this is general economics, resource economics. All right. Actually, all that I want you to look at is if you read from here, if you just start from somewhere, you see, if you start from somewhere here, the net present value, was used to compare, and then but from birth until the end, you will see that the person is arguing and giving reasons why they feel that this methodology fits their research. So this is a classical example where you can plug a method, and though the method is well established, but your work is so unique that you feel that this method fits your work, then you need to convince us why you think this method is good for your work. So this is exactly what they did here. So that is what I just wanted you to tell me. So you can see here, apart from the highlighted runs, where you got the clue is from BAT. Then at the end of the day, apart from until the end, these are all to tell you that, look, this method suits our work very well. That's why we are adopting the method. That's all. Now, what about this? Well, again, if you read it carefully, this is a method that has been modified from a ratio of 1:3, and they are using 1:10. So now this is a standard method, and then you are bringing it to 1:10. So once you change the ratio, you have to tell us why you change, and you need to convince us. And you can see from here the reasons behind the modification. So this is a way of modifying a method. So you can see here, this is the modification, and we have been given every reasonable reason, very good reasons for us to accept that, yes, what you are talking is logical, and you may go ahead, and we believe that you are not contravening any kind of fundamentals. So these are some of the things that I can share with you. Now, what about this? Now, for this one, this is what we call a standard method. So if it's a standard method, you just reference them, and you don't need to describe too many things. So here you see selected chemical properties using what? Standard methods. What are the methods? Then this is the method, this is the method, this is the method. So to summarize it, what I'm trying to tell you is, in scientific research, there are 4 ways of trying to find methods. One is when the method is not a standard one, and you want to write on it and then give value to it. Number 2, the methods are already standardized and you are using them. So here you don't need much description, so long as they suit your whatever your research. The third one is you are adopting a method and you want to justify that they really fit into your work. You see, the example that I gave you, they are working on agricultural waste management, but they took the resource economic type of viability and brought it in. So they are trying to tell you why they think that NPV meets the requirement. And the last part that I'm saying, the third part is what? Where you are trying to modify. So once you modify any part of the research and method that you modify, you will have to explain, and you must convince us that the method is worthy of listening to and is worthy of using. You are also contributing. And the last part is the standard methods. Now, what are some of the common mistakes? Remember here, I'm not sure, maybe this is a little bit controversial. I read one thesis. It's not in the sciences very well, but I can see that a lot of background information again about the methodology. A lot of background information. I think background information, more of it have already been covered in your introduction part. So don't bring introduction part into the methodology. We don't do that. It's very, very important, because each segment has its own place. But if you just want to introduce 1 or 2 lines to bring us to the realm, then that is fine, right? So example, look at this. What do you think about this example? It says samples were prepared using the method described by Newton et al. 2000. You see, and then you are saying that this theory was first proposed by Newton. This part to me is irrelevant, because we already know. So this part it is very irrelevant, because even if we need it, it could have featured in your introduction or maybe in your results and discussion part, but actually this is not necessary. So you just go straight and tell us about this. So that is what we want, right? Now, also, if possible, it will be very important sometimes to use diagram.
Yeah, Prof is there.
Make it easy for us. So example, this description that you see is about this. It's about this experiment. And all that they are trying to tell you that they put some fertilizer here, they blow gas using aquarium pump, and then the gas, which is a greenhouse gas, will get into this beaker, and it has a boric acid, and it will capture, and then that is it. So if you want to describe the whole of this, who is going to understand this? That is why it's better you show a figure or a diagram or a flow chart. It's very, very important in the methodology. You can imagine you want to write this in a textual form. That will be very, very difficult. So once you give it this way and you just give the point, okay, this is what I mean. This is my main drain. This is my secondary drain. This is my field drain. It makes it beautiful and straightforward. But if you were to describe it, this will be very difficult. What about this also, if you want to describe? So the point is where you make the work as simple as possible, but you are able to communicate the information effectively. Now, be very, very professional. Be very, very professional. Again, because most of you are coming from different backgrounds, maybe some of the things you might not agree, that is fine. But what I do know is wherever there is a source of error which is not mechanical error, it is not a place that you need to discuss the source of error in your material and method section. That is not the place. You will have to discuss this in the discussion section. That is what the discussion session is cut for. So these are some of the things. You see some of these things and it's very, very irritating. Look at this. Just look at this and then tell me what I'm trying to say. Look at this text and see whether you can figure out something. Maybe this is very short one. So let's say 1 minute, oh no, 30 seconds. I'm giving you short things so that you don't get bored. Okay, during image stretching, some resolution was lost, possibly interfering with count of craters and whatever. You see here, if you are very fast and sharp, you will see that from here, possibly here, sorry, possibly interfering, whatever. This part is actually discussion. You are interpreting. So here in methodology section, you don't interpret anything. So here, this possibly until here is irrelevant. You don't have to include it. So when we say methodology, just tell us what you have done. During image stretching, some resolution was lost. That's all. The rest of the explanation will come later. Okay. Now, let's go to the results section. When we talk about results, you are only presenting important findings without interpreting them. I'm not sure in your field, but what I do know is results section is just telling us what you have obtained. What the data means is the interpretation section that is called discussion. Unless you are publishing your manuscript, you are following a format where results and discussion have been merged. If they are separated, you have to separate. But if they are merged, they go together. Then once you give the results, you follow by the interpretation. In that case, yes. But if we have already segmented them, then you don't need to discard the result. Just give it the result, that's all. And rather, you should focus on, you have to extract the information, the important information, and look for trends, pointers, indicators. so that when you go to the discussion session, you should be able to do that. This is why you will have to convert your raw data into things like what? Text, tables, figures, plates, maps, photographs. This is where you have to get them in. But I'm going to talk about these things getting to the end of my talk. Right. Now, also remember that you present your data in an orderly way vis-a-vis what? Your methodology. And remember your hypothesis, your objectives, and the questions that you are trying to work on in an orderly manner. What I mean by orderly manner means from what? Important to less important. From simple to what? Complex. That is what we mean. Now, whatever you want to do here, again, it depends on the way you look at it. But generally, you will see that the general style of writing in results will be in past tense. If you want to write in present continuous, you must go ahead. But once you miss it, then you will be heading for trouble. I'm not saying it's a must, but generally that's how it is. Now, remember, tables that you use should be well set out, cross-check them, and make sure that they are okay, but do not discuss them yet. The next part will be also, this is very important, long list of numbers. I'll show you very soon. Let's say, example, you have done a research and you have a table and you have given the values within the table. And in the text, you are almost repeating the values in the table within your text. That means that the same data is in a textual form and the same data is also in a table form. This is redundant. You only have to highlight the most important figures or numbers in the text. That is fine, but not all. So it's very, very important that you should do that. And if there are too many of them, just say the selected example, the selected physical properties of these are summarized in table 1. Then anybody who wants to go there, then you'll be referring to table 1, the important points. That will be very good. And you can only do this if you write in a very simple, short and sweet manner. I have already given you some of the examples in my previous lectures. So there will be no excessive words. In the results section, we don't need excessive words. It's yes or no. Why this is different from that, that's something else. Now, example, when we say excessiveness, it is clearly evident from picture 1 that bird species richness in, no, this is too much. It's not necessary. All that you can say is bird species richness increase with habitat into bracket figure 1. Sometimes it's not necessary that you should even. When you say it is clearly evident, this is very irritating. You are telling people that they are dummies or they are not clever to see. So that is not a good way to write, right? So the simple way is just like this, right? Now, if possible, sometimes you read certain, papers. It's like you are reading a statistical paper. A paper that is fully on statistics. If you are just applying regression, there is no need for you to start to explain and tell us what regression is. That is not the point, unless you are adding on some component for regression or whatever, that is it. You see, if it's ANOVA, this is not the place for you to tell us what is ANOVA. Just take them briefly. Why you use ANOVA regression is enough. R square, we know what is R square. What is R, we know. This is not the place. Explain later in your discussion exactly what you found. That is very, very important. So here, example, you can see, if you read here, this is an example. They just tell you and they say there's no significant difference. Fine. You don't have to tell us, oh, I use ANOVA, oh, I use regression, and you know ANOVA is likely. No, we don't want to hear this. We already know this. And also, if possible, be professional in reporting. Be very, very professional. Sometimes, example, I read one thesis again, and some of the models or some of the predictions really did not actually work well. You get what I'm saying? 1 or 2 or 3 did not fit the what they call the hypothesis, and they were credible enough to say that this part did not meet our expectation. This part is okay. That tells us how credible you are, and somebody can take it from that end, or you can even improve in your subsequent research. This is very, very important. There are some of us that when we get negative results and they are contrary to what we expect, then we want to what? Suppress them, or we want to hide them. That is not really a good thing. Negative results also should be explained if possible, right? So here, this is an example. You conducted a research and you said that you find out that some of your plants died or something happened to your plants. Just state why they died, as simple as that. You have a reason. Here they want to tell you that the pH is above 6. That is why they die. Or maybe the salinity might be high or whatever. Just say it and explain. Unless it's a mechanical error, there's no problem. This is a classical example of a research by one of my former PhD students. You see here, we use different amount of organic fertilizer. And then we realized that we thought that setting optimum would be better. But when we exceeded the optimum, you see the plants start to die. You see the response started responding, and here they die. So we have to explain why they die. And if we are going to hide this and not reporting, actually we are curtailing the advancement of knowledge. At least we should be able to explain, so that if somebody is repeating, they know that this part is not good, we shouldn't go there, or we should find a way to overcome the problem. This is what we call extension of knowledge, right? Now, if you are dealing with symbols, you are allowed to use this sign. This sign is approximately. So if you are using symbol, it's allowed. But if you want to write it in English, then you say approximately is also okay. Remember, approximately is completely different from about. Approximately is more formal and more precise, right? Now, also remember, if possible, do not start a sentence with figures or numbers. Create the sentence in a way that you avoid the numbers, especially when you have more than 99. You can imagine if you have 9,195. Are you going to write this as a sentence? So you need to know the style of writing so that you avoid it. This is an example. You see, if I were to start, I'm going to say 500 grams of soil. But if you know the style of writing, only using A here already qualifies, so it saves you from spelling the whole of this. So this is the beauty of knowing how to write and juggle your way through. It's very, very important. Only a letter has saved you all this, right? And these are some of the examples. So here, this is why you have to know how to use A, N, and then V. Is very, very important in writing. So once you know how to write them, then they are going to save you a lot. And this is a very simple way to see whether somebody has control over the English language, especially in technical writing. Also remember, 1, 2, 3, 4, 5 until 9, whenever you state them in a text, they must be in words. Never say 1, 1, no. You must spell 1, 2, 3 until 9. From 10 going, then you give figures. That's the way it is done. And also note that when you give a unit, the moment you use A, then you are giving a unit, then there must be a dash between the number and the unit. But when you don't use A and you use 8, then it should be what? 8 billion. And even this is wrong. You must use 8 that is spelled 8 billion, because this is less than 10. If it's 11 billion, 10 billion, then you write a 10. These are some of the examples. Also remember your units. These are these are not forgivable. If you are talking about units, certain units, the number and the unit, there will be no spacing. So here, example, meters, kilometers. Here you need to give some spacing. But if it's percentage, there's no need for you to give any word space. Very, very important. And also, just like last 3 weeks, I did say, wherever you have numbers and the number is in thousands, remember every 3 words there must be a comma. Every 3. So 1 million, you have comma here, 10,000, comma 1,000. And remember, when you are reporting 0.87, not 0.87. Again, one thesis that I read is the same. They just give the point. No, that is not a standard way of writing. Very, very important. And then we also have to look into this. Decimal points. We have to be very, very careful. You see, when you look at here, we can write this, is acceptable. And if you write it this way too, it's acceptable. Why? Because here you see the decimal point here is 1. Here is 1. If you approximate this is 12. And approximate this is 8. So that is fine. But you cannot approximate like this. This is not possible. Because if you approximate this, this becomes a whole number. And this will never become a whole number. So we have to be very careful here. And then the next part will be, it's very, very important that when you are dealing with standard deviation or standard errors, I can see that some people give a whole lot of decimal points. Only 2 is enough. If you are talking about standard deviation and standard error, the implication is if you give 0.0009, it doesn't exist. So 2 should be enough, right? But if you are talking about descriptive statistics, example, you are talking about height, weight, and whatever. If precision is not very important, then you need only 1 decimal point is enough. Unless you want to be very, very precise, then that is something else. And also it depends. Let's say you are talking about weight. What kind of machine did you use? Maybe the machine has only 1 decimal point. You have to report based on 1 decimal point. You cannot report based on 10 decimal point. It doesn't exist. So these are some of the things that we look for. And when you are talking about your relationships like correlation, regression, you will see your R, they write a whole lot of numbers. Actually, only two decimal points should be enough for your R and your R squared. 2 decimal points should be enough. What are you trying to prove actually when you give more? It doesn't make sense at all. And then also remember percentages. If you are dealing with percentages, it's very important that you go up to 2. But it depends on your scale anyway. If the scale is very, very sensitive and you need detail, then you have to go to that level. Now, probability level. When you are using probability level, we have something called P less than, P greater than, P less than or equal to, P greater than or equal to, P is equal to. You have to understand all these things. But the most standard method that is quite precise in relation to your standard errors will be the P is equal to. If you use that one, it gives us the precise probability level. Because when you say P less than, it's a range. If you say P greater than, that is fine. And sometimes when you have no significant level, we know which P to use. When there's significant level, which. So this kind of interpretation should be very clear to you. So this is why when you are using the annotated kind of thing, if let's say you produce a table or a figure where certain variables, there is no significant difference, certain variable, there is significant difference, then you can use P less than because some are not significant. So these are some of the things. Now, when you are talking about consistency in terms of standard error, standard deviation, and the like, what do you call it, measures of dispersion, then you need to understand this. You look at this. 80 plus or minus 10 is correct. 0.040 here, you see that you are matching the what? The decimal point against this decimal point. But here, you will see that even though you have 3 numbers, you are having only 1 because if you put 0.0, it doesn't mean anything. 0.0 is 1 digit, but we don't write 0.0. This is why you leave it 180 plus this. So these basic things must be very clear. We don't write like this. They are very, very important in writing in very scholarly journals, very recognized journals. These will gives you some class in your writing. And then normally confidence level. Sometimes we see that people just misuse confidence level. Confidence level depends on the nature of your work, of your research. If I'm dealing with aviation, where precision must be at the highest level, then my confidence level will be maybe 99.9%. Or I'm going to perform a surgery. Or you are talking about going to the space. You see, these kind of research, we must have high precision level. But let's say if you are dealing with, let's say like human beings and you are taking about interviews or whatever, or you are dealing with field study, and you are telling me that your results, you are very confident, 99.99, we know you don't know what you are talking about. If it's a field study, there is a lot of what we call it, extra, interferences that you're unable to control. So please know how to use your alpha level. Is very, very important. And then also, when you are explaining statistical findings, you compare this relationship with that, and you say it is highly significant, or there is a high relationship, or these kind of things, if possible, tone down, because you are dealing with error level. Try to be moderate. Even if you get your relationship to be, let's say, 0.97 or 0.99, tone down. Just say there is a relationship. Whenever you want to say a strong or weak or whatever, these are all subjective. You get it? When you are in the secondary school, they say when your R square is less than 0.05, then you say it's weak. If it's getting to 0.9, it's strong. These are all relative things. So because we just want to see your error level. What about if your R square is good and your standard error is not good? So we need to be moderate in the way we talk about things, right? So my next point here that I would like to share with you is your error bars, credibility in your work. Your error bars must be very, very careful. If you present error bars like this, it tells us that your work has no credit at all, because it's suspect. You have a lot of errors. These are your standard errors. How can your standard error be half of your mean? That means that something is wrong. So these are some of the things. This data will never be published in any serious journal. And also, we will also have to know that you have to understand that never say, don't contradict yourself. When there is no statistical difference and you see that one value is higher than the other, don't say, even though there is no difference, maybe my research A is better than research B. Why? Because the mean A is higher than mean B. That is contradiction, because the difference you see is due to statistical error. So you cannot accept that kind of whatever. So don't contradict yourself in terms of that. And it tells us you don't know how to interpret statistics, or you don't have a good understanding of statistics. Now, the next part will be, what are the common mistakes in results? Common mistakes in results is that do not include raw data in your results section. Raw data is never communicated because the value is very low. So present the key results that are very relevant, and they are hinging on your question and the problem that you are trying to address. Now, if you say you will still want to present the raw data, then it will be better you present them in a form of mean, standard error, percentages, standard deviation, CV, and whatever. Actually, there is meaning to that. Your range, upper quartile, lower quartile, the skewness, kurtosis, then you can go into these things. Fine. Now, it's very important that if you have decided to write your information in table form, then you cannot repeat it in a figure form. Later, we are going to talk about when to use table, when to use figure, photographs, and whatever. I'll be talking about them. So you use text to clarify. You use figures or tables to summarize your data. Very, very important. Now, here also, sometimes it's not always good that you need to repeat all the figures, numbers, or whatever that you have in your tables. Once in a while, you can do something like this. But to me, I would prefer it to be like that. I've given you already the category in the table, then I don't need to repeat the values. But once in a while, if you repeat the values, there is no harm in that. It's acceptable. But it shouldn't take a chunk of your the paper. Now, when you come to interpretation of your data, please, it's very, very important. Remember, in the results section, you don't discuss your data. You don't interpret. You are just doing comparison, or you are just looking at the meaning and the trends. But when you come to discussion part, this is where you need to show your understanding and your level of control of the subject matter. Now, you also have to understand that when you are in the results session, it's not important that you should discuss your source of errors. Your source of errors are discussed only in the discussion level. So we will talk about that very soon. So this is an example. The selected chemical properties of this are typical of this because of fertilization. This is wrong. If this is in the method in the resource section, because of fertilization is more to discussion. So it cannot be in the resource section. It must go to the discuss section unless you have combined the two. Right. Sometimes also with the section and certain professions like mathematics, you are talking about engineering or whatever, it's very, very important that you must have what? At least some results. Figures and tables are a must in any manuscript, unless you are dealing with certain professions like example, mathematics, computer science, or whatever. But generally, you must have figures and tables, at least in your work. And then also, it's very, very important that if you are reporting information on material and method, you shouldn't actually make sure that you are dealing with the right reported. It's very, very important, right? Here, this is what I mean here. Sometimes you have got background information. The background information, normally, if it's a background information, I don't know your area. But in our area, if it has nothing to do with your research and it's just a background information, it is reported right under the methodology. But if you want to show us it's part of your work, then it comes to the discussion section. Here, this is what I'm saying. In my field, this is not part of my research. It's just characterization. I will send it back to the methodology. Right. Now, what do you think about this? We found out a sample contained what? Pyroxene. So a group of sample B, whatever. If you look at this part, what I can tell you here is this is the only resource part. The one in the black is the only. The red one is never resource. It's about discussion. So methodology. So it must go back to the methodology, and you don't put it in your resource part. So this is what you should write. We found that sample A and B contain pyroxene. That's it. So this red part is a methodology. Why? Because you say that what? The powder and whatever experiment will run again. You run again, then it must go to the methodology. Now, it's very, very important also that we talk about this area. Orderliness that I was talking about, what I meant was you should start from the most important part of your research and end it to the least. This is why sometimes you have your primary data and your secondary data. Which one is very important for your work? And then when you start to present your data, present it from simple to complex. You don't start from complex to simple. It's very, very important. Example, if you have this data, for instance, it will be very, very important. If you say pH, total carbon, there must be a reason why you arrange all these things this way. It's not for fun. And there must be a reason why you also went horizontally. It's very, very important that these are very clear. You don't do things anyhow without any knowledge. It's very, very important. This is where people are going now. So these are some of the things that I could share with you. Now, the last part before I go into the interpretation part is extra words. You see, every stage I've been emphasizing that avoid extra words, avoid extra words. So when you come to resource section also, example, it is shown that in table 1, X induce Y. You see, you can write it in a very simple way. This it is shown is not necessary. Just say X induce Y into bracket table 1. That's all. So it makes it very simple and very beautiful. It is all these kind of things, you just forget about them. Again, like I said, use past tense to describe what happened, especially even though it's in the resource section. If you are explaining something, use past tense. So this is why you see, you can read this later on and you will see what is going on here, right? But here, what I would say here is example. Do you remember I did say that if, let's say, you have already chronicled all these things in your table, why do you go back to repeat almost all of them? So if I were you, I will leave them in the table and I'll start to refer them to, unless I'm concluding, then I'll give some key values or in my abstract. Otherwise, the table is enough for us to refer. And these are some of the examples. Again, if you look at here, this is a resource part plus methodology. Further temperature increase were avoided by turning and mixing the compost. This is methodology. It shouldn't come in your what? In your results. Now look at it. As has been generally observed. Here you are talking about what? Discussion. So this should not be here. It should be in your discussion. So the black one here and there, these are the results. So focus on those kind of results. Unless you have bring them together, results and discussion, then that is fine. Right. So these are some of the examples that I think if you have time, you may go through and then you will see what I'm trying to say. Right. Now let us go to the main part of the discussion and then we see. When we talk about discussion part of your paper, here we are talking about interpreting your findings. So now you are drawing out your main achievements and then you explain them. So that is where the whole argument will start, their intellectual ability, the deductive reasoning, and all sorts of things, inductive or whatever, this is where they will start. Right. Now here, this is also where you make links between your objectives and your findings, and also make links with the literature. This is where you demonstrate that kind of flair, the art of your understanding, and what we can get from your work. So here, interpret your work as much as you can in relation to your objectives. As you move on, ask yourself, am I discussing my work in relation to my objectives or not? If you are outside this scope, then it's just a bluff. So we have to know that. And you yourself should also relate the work to your knowledge by going through the objectives, but you also have to relate it to what others have done. And you have to be very careful here. If you consistently say that my work is consistent with others, my work agrees with this and this and this and this, that means that there is nothing new about your work. Of course, yes, we know you want to establish that, yes, you are in the right way or whatever, unless you are comparing indices. But it will be good that you also highlight something new about your work or something that, it try to, contradict others or is not consistent. That blows out your contribution very well and it comes out very nicely. Right. Now, remember, it's very, very important that we look into this aspect. When you are interpreting your data, ask yourself these following questions and whether you are addressing them or not. Did the results provide answers to your hypothesis or not? If yes, what is the meaning? If no, why? Very, very important. Now here, number 2, if no, can you suggest alternative hypothesis? That means that you keep discussing or whatever, but in another realm, if we were to repeat your work or somebody wants to take your work on, can you suggest something? Example, what is the hypothesis and why? What is the new one that you think in subsequent research could be taken care of? Now, or in your own research, do you think that we could do something to consolidate the new hypothesis that you are trying to find, or how you deviated from your own hypothesis, or it just does not follow natural order? And it's also very important to know whether the hypothesis is new. If it's new, that is a credit. If it's not new, are you just confirming or not? And even if you are confirming, are you adding on to knowledge or not? So these are the questions that as you discuss your work, you should pull them out, and any reviewer will be looking for these things. And then, are there any biological principles, theories, concepts that you are trying to establish, or you are just reinforcing? So these are the realities that we need to look into. Now, your results, are they in agreement with others or not? If yes, do you have, even though they agree, do you have a better explanation? If you have a better explanation, that would be fantastic, because now you are making us to understand the area very well. If you are very, very, in touch, in tune with Nobel Prize winners, you hear that whenever they give them the prize, they say that his work or their work help us to understand or help us to explain You get what I'm saying? So if that is where you are, that is very good. If no, how different is yours from others? Maybe you are come out with something new that nobody thought about. Fine, that is good for you. But be careful. You are deviating from others because there are some flaws in your work, and then you are in trouble. Then that cannot be accepted anywhere. So those are the things. Now, the next question that you need to ask yourself, can you draw some general issues from your work or not? Can you make some generalizations, and are they grounded in any scientific, background or whatever? Or do you have any theoretical or practical way of working on it? These are the things. Now, this is where you explain the sources of error. These sources of error, I'm not talking about intentional error. I'm talking about errors within the realm of statistics that are acceptable. Now, how were they affected? Was it because your sample was not taken very well? Did you have problem with your, or did you also have problem with your, just like your analysis was not robust enough or what? So we will have to know. And then, do you have anomalous data? Very strange data. Is it really a mechanical error, or it is just a data that we never thought about that we are getting? Or do you have any theory to explain? Or is just a very serious error that has nothing to do with your work? Example, instead of writing 16, you wrote 61. And that is a serious error. It's an anomaly that cannot be fixed unless you repeat the work or you cross-check your work. Now, the next part, was your study the most effective way? Are you really sure that the experiment that you conduct or the research that you are communicating, you have exhausted the area and you are talking about the credibility of your work is all right? If no, how could it be improved? These are some of the things that we need to take into account. Now, remember, you yourself, at the end of your work, if your results and your conclusion did not help you to understand the problem, then something is wrong. And this is where most PhD candidates fail. They themselves could not understand the problem. They are not confident. They don't have the knowledge of what they are presenting. How dare you going to get Doctor of Philosophy? So if you yourself don't understand, forget it. Now, can you propose other studies that you think that could reinforce the area? That is your contribution. And any further studies that could be done must have hypotheses. It's very, very important. And this hypothesis, can it be disproved or not? Can it be falsified? Because if it cannot be falsified, then it's not a good proposition. So we have to know that aspect. And most of the questions that we have been talking about, you must have evidence. It's not like I think. Not too much speculation. Once a while is okay. By deduction and induction, they must be grounded. We must know that. Now, do not over-conclude. If your work can solve a problem, stop there. Limit yourself there. Don't say it can solve A, B, C, D when it is not true. It's very, very important. Conclude based on your results with vis-a-vis your objectives. These are the things. Now, speculation is allowed, but it must be very moderate and reasonable. It's very, very important. The moment that your speculation is too much in your discussion section, what it tells us is that you don't have enough data. You couldn't generate enough data to really, really answer most of the issues. Or you just don't understand the area that you are working with, or you are not abreast with time, you don't have in-depth knowledge in the area. Now, it's very, very important that when you are ending your discussion part, try to wind up narrowly, like you are bringing up a whole sort of summary. It's not the conclusion section, but as we are getting to the end, you should bring us back for us to know. You have talked about so many things, but these are the salient things or the important things that you are drawing our attention to, and they must be very, very significant. Otherwise, we get lost, you also get lost, right? And remember, as you discuss your work, always remember your objectives. Your discussion must be hovering around your objectives. Otherwise, you will never be able to conclude very well. Now, this, and then also hammer, try to stress a lot on the significance of your work and how unique your work is, but don't overdo things. We don't overdo, please. Right. What are the common mistakes? The common mistakes could be, one, if you are publishing a paper or you are sending your manuscript and you have been asked to separate results from discussion, you must do that. Otherwise, once you produce your results and you follow by discussion, is outright rejection because you have not followed the what? The instructions. And I can assure you that, to be very honest with you, now most of us are going that direction. Because sometimes when you combine, we don't see the relationship very well. So once we separate them, then we can. But I tell you, if you separate results from discussion, it's a little bit difficult to write than to go straight and merge them. But again, because we want to see the clarity and how results relate to discussion. So these are some of the things. If you have time and you read, you will see the first one is more to results and the second one is more to discussion. This is a combined type. If that is what they want, you go. Right. Now, remember, results section is for facts. You are presenting your facts, and discussion section is for you to interpret your data. That is the major difference between results section and discussion section. Now, also be very careful. If you have not reported any results that in your results section, and all of a sudden you bring new results in the discussion section, that is not acceptable. You only discuss what you have presented in your results section. If you want to bring a new thing, you have to go back and put it in the results section. From there, you discuss. That is the way it is done. And then we must also try as much as we can to avoid sweeping statements. Broad statements. It's like you have just conducted a research and it's a very simple research and you say it can solve the world problem. That way of doing things. Major breakthroughs are for groundbreaking research. Groundbreaking research are the one that get this. Yours is just a small part, just narrow yourself like that. So remember, yours is just a tiny part and then we have the bigger picture. Now, never end your discussion without having some conclusive statements. If you don't even have conclusive statement, just say something for us to see that even you couldn't achieve what you wanted to achieve. Say something for us to see that there is a direction, there is a reason. But you just leave it hanging like that is not acceptable. And in the course of writing, interpreting your data, avoid ambiguity. Discuss your work as clearly as possible so that other authors can understand your work. And avoid missing information. Example, no reinstating of hypothesis of motivation. Once a while is okay. But sometimes you also have to relate your work to the bigger field. Remember, you have a theoretical framework. That theory has already been established. You need to be relating to that. It's very, very important. Sometimes also you just forget to compare your work to others. And the last part is you never discuss your source of errors. It's very important to add credit to your work. So these are some of the things that I can share with you. And then when you get to the concluding part of the manuscript, this is where you draw your arguments and then your findings together. You are closing on to your objectives and how they relate to your conclusions. And now, you have to leave your readers with a very strong sense that your work set out has done what it's supposed to do. And it is very, very important. No exaggeration here, but you have to have a very strong statement. Yes, you have been able to solve the problem. It's just an example. Is this lecture boring or not? No, it is not boring. Yes, it's boring. That is the conclusion. It's a very strong word. So it must summarize your major findings. And if you have limitations, the limitations must be there so that people can take it on. Now, present implications if you can is very, very important, and suggest further what? future direction. If you can do that, that will be very good. Like I said, end your work in a very strong note. It's very, very important. Now, once you get to this part, remember, conclusion part, to summarize it all, is where you give the take-home message. And your take-home message must be very clear and must be concise. And it must be done in accordance with what? Your goals or your objectives. And remember, never, never end any manuscript without a conclusion. It is never done and it is not acceptable. I have seen a journal where they don't have conclusion section. By getting to the end of your discussion, they will compel you to conclude there. There is not like a caption for conclusion. So you have to understand it. That is what I mean. So these are some of the ways that you conclude. I don't have to go through this. And then now, recommendation is very important. It's not a must. But if you can provide recommendation, it's very, very good, because you are paving way or you are giving some directions that people can go into. And I'll share my personal experience with you. So it provides focus for future activities, and it also ensures continuity, you know, so it doesn't end there. So it keeps moving. I mean, that is science. Science is dynamic. You remember, academia is very dynamic. And as you are giving such, recommendations or whatever, being it positive or negative, give. Sometimes there were certain things that you couldn't achieve. It's okay. So long as the fundamentals have been achieved, that could be another way of giving others way to do some work. It's very, very important. Then I'll share this for you to see. This is a research that I think during my postdoc, I think this was a research that I did. So this is part of the recommendation that I gave. So after a few years, I received an email asking me whether these recommendations that I gave, if I'm yet to pursue them, I think this came from US, they want to take it from there. And I wrote back to them and told them I couldn't continue, but if they want to continue, that is fine. So this is the beauty of what we call the academia. Now, what about your references? When you come down to your references, remember the references are very important, because it tells us that you are in tune. You know the story. Where are we going? You got what I'm saying? It's very, very important, and it adds credits to your work. And remember also, depending on the type, whether it's in alphabetical order or numbering or whatever, you will have to follow based on the, what you call it, the kind of journal that you are dealing with. And sometimes some of them could be annotated. So it's very, very important that you know. And it's very, very important that if it's a secondary reference, unless you want to say adapted from something and you have the primary reference, it's better you go for the primary. That is very, very important. Because if you also give credit to others, is very important. What about if you go to the secondary and then what we call it, the quotation is wrong? Then it becomes a problem. Remember, plagiarism also comes in. And of course, when you have the references also, it helps us to have the background of the information to understand your work and also contribute to the context of your work that you are talking about. That is fine. And remember, the bulk of your papers should come from peer-reviewed papers, not just ordinary textbook, simple reports, or maybe things that are not very credible, newspapers or whatever. No. I read one thesis and they were having newspaper. I had to tell them that to get away with the newspaper report, because we are not sure about the credibility. Now, that is what I have just said here. And also, because now we are talking about H-index, citations and whatever, please do not cite your group or your friends because you want their citation index to increase. That is very unethical. Don't, even your own work, if it has no relevance to the paper that you are writing or manuscript, forget it. And then reference section, if it's your own work and it's about 50% referencing your own work, I think that is not acceptable at all. And you also don't have to force your student to be referencing you because you want high H-index citation to get promotion. That is very unethical and it's academic dishonesty or academic fraud. Now, if you remember, if you realize, I pull out data presentation under results section. So now I'm bringing back the data presentation and then I'm going to conclude. So here, data presentation, from all the noise that I've been making, if you don't present your data very well, then you are unable to communicate your information effectively. So it's a very powerful way of trying to communicate your work. And it can come in different forms: text, tables, figures, map, plates, and whatever. Now, for you to do a good job on data transformation, you have to know what type of variables you are dealing with. Are they quantitative, qualitative? Within quantitative, are they continuous, discrete? Are you talking about ratio, interval, ordinal, nominal data? Because by knowing this, you will be able to know how best you can produce your information and communicate them effectively. So it's very, very important that we take these things seriously. Right. Now, this is very important again, because when you have a very large set of data, which not sometimes it will be very difficult for you to write them in text form, and you don't have a choice to communicate them effectively, you might need something like tables, figures, graphs. These are very, very important. And for me, this is very, very serious. This is on data analysis and presentation. It's very, very important. Right. So what we need to understand here could be, when you display your item, you have to clearly show the most important findings, and they should be standalone. What do we mean? For instance, if you give me your table or a figure, and you give me the caption of the figure and the table, if I don't read the text of your paper or manuscript, your table, the content and the caption should help me to understand what is in your table, even without backing with the text. If you can do that, that is what we call standalone information, and it's very, very good that you should inculcate this. Now, also, some readers, especially I've seen some editors, I have seen some reviewers, some colleagues, if you give them your manuscript, what they do is they skip almost every part, and they go for your the way you present your data. They go look for your tables, go look for your figures, your plates. Those are the things that they go look first before they even come to the body of your work. So it's very, very important. That's why when you are talking about tables, tables are good for when you have very complex data, very large data, that you cannot manage them in textual form or in figure form. Then table could be the right place to put them. Figures are very important for when you want to make some convincing argument, but through visualization, so that the moment you project the picture, we could see trends and we could see what you are trying to say without too much of argument. Then that is fine. Now let us pick them one by one then, and let's discuss them. What is the place of a table? Table here contain a list of numbers or text in columns and in rows. That's what tables are for. But remember, tables are never used to show relationships. Tables are used to show differences. So when you are comparing certain things and you want to see with some quantitative figures, values, then that is fine. If you want to see trends and relationship, then you have to go for figures. Now, what we say here, sometimes also, you might have a very complex data that when you present them in a figure form, the whole thing becomes difficult and messy. Then in that case, you may decide to go for what we call a table. But if it's readable and it's quantitative, then figure will be a good way to do things. Now, this is an example. So here, if you have a table and we are interested in the exact values and whatever, so to present the whole of this information in a figure form will be very difficult. Therefore, you will want to present them in a table form with the variables plus the spacing and the unit, and we see the exact figures, then table could be a good place to go. Now, these are some of the examples. Now, when a reader needs accurate information, remember when you use graphs, when you use photographs or whatever, they just give us the idea, but to get the accurate information, sometimes you have to extrapolate unless you put the the figures inside. So here, this is what I mean. If I want to know pH, I know this is 4.81, but if I use a bar graph, maybe I have to extrapolate and I may not get the right, figure. So in this case, if that is what you want to do, then this is the place. Now, sometimes also you use tables when it's going to be very messy, when you use your graphical form, as I said. I'm just magnifying it again. How are you going to present the whole of this in a graphical form? It becomes very difficult. And some more plus what? Standard errors and whatever. That will be very difficult. And then sometimes also certain journals will compel you to change your figures into tables because of space and then the cost involved. Sometimes they tell you only 10 pages. If you want to go more than that, every page you have to pay. So in that case, you are being compelled and you don't have a choice, you have to follow. This is why sometimes it's very important that if you have your table, you do not include any table in the text of your work without discussing it clearly. Very important. If you have a table, you have to discuss the table. Now, place a table immediately after a paragraph. You will see that it's very, very strange that when we are writing a paper, you will see that you bring the table first, then beneath the table, you start to write your text. Actually, that's not the right way. Of course, in general, they do, but in reality, the text must come in a paragraph, then you refer to the table, then from there you continue. Now, also remember that the title of a table should always be on top and it should not only be below. It's always be on top of your what? Table, the title. Now, the title of a table must also reflect the content. It must be very informative so that even with the title alone, we don't read the text. It should give us a very broad overview about the content of your table, what is in the table. So it's very, very important that we must understand this. And any good table must have at least standard deviation or standard error if it's quantitative. So you can see here, you have your standard deviation and standard errors. And then getting to the end, a good table also must have what we call category. It's a category that is in row and column. A good table must also show spacing, the spacing between rows and column. And it must also have a well-defined unit. Your unit must be very clear. And then the font type and the font size must be they must be readable. And then your legend or the captions must be very, very clear. Now, what about some of the deficiencies of table? Tables sometimes can take a lot of time. You have a huge data and it take you a lot of time to describe and whatever. Sometimes it takes time. But if it's a graphical form, in a visual way, we can understand what is going on. And sometimes they are not easy to communicate, especially when you want to point out trends. It could be quite difficult. Sometimes it's very compelling that figure. Here you can see that compelling reader will find it difficult to find out the trends. Even you yourself, if you are finding it difficult. What about those who are new to your data? So those are some of the things. Now, when you talk about data also, we have them in plots. So here I'm leading you into what we call figures. So we call them data plots. Now, data plots, normally, they will quickly show you an information that will be in quantitative terms. So if it's in quantitative terms, you see sometimes you want to see a functional relationship or statistical relationship between 2 or 3 variables or whatever or more, then this is where you could count on your data plot. Now, data plot also can display data in an objective way, but we have to know whether they are discrete or they are continuous. So if, let's say, for instance, they are discrete or whatever, we have to know what requirement are we basing on. It's very, very important. This is why we should know whether the axes are properly labeled or not, and then we have clear units. If you want to go for figures, your axis must be properly labeled and you must have your clear units. Now, you must also define your legends in terms of error bars. We must also define the annotations, your R-square equations, very, very important. And the font type and the font size must also be very, very clear. Now, if we talk about data plots, there are 2 types. We are talking about discrete and continuous. Now, when you talk about discrete, remember discrete, we can have discrete that is qualitative, discrete that is quantitative. For the sake of this talk, I'm talking about discrete that is quantitative. Here is a categorical data, but you don't have decimal points and it cannot take all possible values. For instance, if you said 10 plants per hectare, 20 students per class, you cannot say 20 and half. Who is that half student? So this becomes a discrete quantitative, and the way you present them will be different. So if it's discrete quantitative, then you have to use what you call bar or line graphs. That could be very, very useful, because bar graphs are standard way to what? Display your variables, where each bar will represent a proportion of an observation in each category. This is what I mean. Example, you have your questionnaire and then you have set out your for whatever, and you draw your information. You have your category, yes, no, maybe. So you have the proportions. So this is discrete. Then you can use this kind of a bar graph. Now, when you talk about line graph, they are very useful for us to see some changes in the variables that you are dealing with, usually over time. So if that is the case, then this is where you have your lines. Then it becomes more or less like a line graph. So you want to compare, but you can see from each point, how does it look like? If that is what you want to establish, then you go for this. Now, what about continuous data? If it's continuous, it means that we have the numeric. You have the numeric on your X and Y axis, then it's possible to divide them in incremental form. That means that you can take fractions, all possible numbers can come in. Example, height, temperature, weight, and the like. So under this, you should know when to use histograms, dot plots, box plots, scatter plots. Very, very important. They have their own meaning. Histogram is just to demonstrate the distribution of a continuous, and then you illustrate whether you are talking about normality or non-normality, whether it's skewed. So this is an example. When you are talking about this kind of presentation, then this is what it is. You can see this is a normal distribution, and this is a skewed data that is skewed to the right. So this is one example. Now, dot plots are also to show distribution of a data, but usually they are used to highlight clusters so that you can pull out your outliers. So this is an example. For instance, it is a cluster, this is a cluster, this is a cluster. Here also you have your cluster, but this becomes an outlier. And this is very important in data management. So whatever you present, we must understand why we are doing the presentation. Extremely important to know this. And then we have something called box plot. It's also a continuous one, but box plot is more for you to show your central tendencies, measures of spread, and outliers. Example, mean, mode, upper quartile. You are talking about these kind of things, maximum, minimum, and whatever. So when you look at this kind of graph, if you look at it carefully, you will see that the box plot we are here, this is the central tendency, and this is the maximum and minimum. So this one become like they are outliers. So if that is what you want to convey, then you have to let us understand this. Right. Now, we also have what you call scatter plots, but these are the ones that show relationships. Example, you are talking about correlation, you are talking about regression, and where you got to regress them and then see your cause and effect. Then in that case, you are going to use scatter plots. And this is where you have your equations or your R square or whatever. This is an example. So, but this kind of presentation must be quantitative. It's not qualitative data. Now, what is very important is it's very important that in continuous data presentation, you will have to understand the data and choose the right kind of presentation. Otherwise, the same presentation can be presented in different ways and different meaning. So it's very important. You see, this is an example. You have A, B, C, and D. They are presented in different ways. This is showing an outlier. This is showing you normality, bimodal, because they are two here. And then here you see on equal side, one sample is higher than other. But all of these things, statistically, we can mean different things. But in general, this is all that they are trying to mean. You compare group A, group B, group A, group B. So if this is what you mean, better use this and forget about the rest. So these are some of the things that I think that we need to be wary of. So to summarize it all, when you talk about figures, it's for visual presentation to illustrate concept and methods. This is an example. When you talk about figures also, you can use figures to show relationships, and you can compare trends and comparisons. And figures also can be in a form of graphs, maps, images, drawings, and so on. We can use them. But use them to communicate specific information. If possible, if you really want to make it beautiful, you can bring figures in multiple within the same, what do you call it, a page. This is what we call multiple. You can see here, this is 1, 2, 3 different colors. So you can see the indentation, what happened here, what happened there, and within these peaks, what are they? So every stage is telling you, so you can do a very good comparison. Can you imagine if you want to separate them individually? That would be very, very difficult to compare. So those are some of the things, and this is another example. And then you can imagine you want to use this, then that would be very difficult. Now, remember also, each figure must have a number and the title, and then in terms of reference in the text, you should be able to do that. And then, just like I told you, remember, when you are talking about figures, we must know independent and dependent variables that are numerical. When they say numeric, if you look at here, these are quantitative, these are quantitative. So you can use your figure, but this is a line graph. If you want a scattered one, then this is how, numeric, numeric. But sometimes also you will have what we call bar graphs. For one, your y is what? Is non-numeric, your x is numeric. What do I mean? You see here, this is not numeric, rice straw, distilled water. This is not numeric. I'm talking about distilled water as water, not the quantity. But you look at your dependent, your y-axis is numeric. So if it's non-numeric and numeric, you will use bar graph. If it's numeric, then you go for continuous, presentation. So this is where the bar graph, and then now you also have to know when to use bar chart. Proportion. Let's say example, if we want to know in Malaysia, how many different countries from Africa, what will be the proportion of students in Malaysia, then we can use something called pie chart. So these are some of the things. Now also remember, your unit must be provided as much as you can. Provide your error bars, and your error bars must be very reasonable. We are not talking about this kind of error bars. And then do not repeat the same information, text, figures, and table. This is never acceptable, and it is out of place. Now, the last part that I want to say is know what kind of presentation will suit best your research work, and you have to have a reason for it. You don't be like you are in an island, you don't know what to present. It's very, very important that we know these kind of things. This is why you have to get all these things clearly. That the last part will be, what are the other forms? We are talking about graphs and plates or photographs. Sometimes it's very good. Photographs can be used to show the setup of an experiment. If that is what you are doing, this is an example where you have show you a photograph to show the experimental setup. Sometimes also can be done like this. This is one of my students measuring ammonia from fertilizers. So this is the setup you need to show. It's easy for us to see. And then photographs also can be in a form of pictorial form to compare treatments. You want to compare two things, you can use photograph. You can see clearly that this is a conventional type of growing rice, and this is our innovation. So we are trying to say that even if you use our innovation, the soil will become healthy, and you can see the grades are far, far better. So you can use to compare, and by using photograph, and it's very, very important and very straightforward. And the last part will be schematic, where you are showing illustration of flow charts. This will be an example of an illustration. And what do we mean by here? Maybe I would like to share with you the last part. This is an example where when we talk about flow chart and to show the concept on whatever, you can see we are working on some big project. And then here, this is the visualization. You can see here, all of them have their own places. So if you can do this, this is what we call flow chart, a very detailed one, so that every stage that we move, we understand. I don't have time to show you, but I'm just illustrating. So you have 4 major parts, and then you can show the relationship, how they are related. So this is what we call a flow chart, not just to describe without any relationship. So this is what I can share with all of you. And then winding up the work, ladies and gentlemen, what I can say here is, if you really want to have your paper published in any kind of serious journal, these basic things should never elude you, and you have to take them seriously. If a reviewer comes back to you, then your level, your status is being already eroded, and you don't sit down for people to tell you this simple thing. Thank you very much for your audience, and I would like to apologize for the long talk, because today is the last day. I don't want to come and have another section. Thank you very much, Dr. Mutaka. Over to you. My respected audience, I'm very grateful for your rapt attention.
MashaAllah. Thank you so much, Prof. I will now open the session for Q&A. But before then, I'd like to, as mentioned earlier, all the sessions we've been conducted, they are free. So with this, we have nothing much, no material compensation can perhaps will be enough for Prof. So just as a token of our appreciation, before we go into Q&A, we would like to just display a certificate for Prof, a certificate of appreciation to Prof for all the sessions that he has taken us through. So Prof, please accept this small certificate of appreciation from us.
Thank you very much.
Okay.
It's a surprise to me. Thank you so much.
This one is for the session on 27th June, where we began on the academic side. Then the second one for the 25th July. Then the third one on 15th August on the science of research. And the fourth one, where we started this session on the remedies for paper rejection. On 24th October, then we had a master in scientific writing. And today, thank you so much. And this is our small token of appreciation. Thank you so much, Prof.
Thank you so much too.
Before
Thank
we
you
go
all.
into the Q&A sessions, just to highlight, for those who wish to further go in details into the findings, just to revise, for revision, can catch up with Prof on, that's next week, Tuesday at 2:30 p.m. This will be organized by the university, UPM. And then, before we forget also, on, I think, 5th December, Prof, I hope we will now perhaps divert into agriculture on World Soil Day to have a session.
Yes, yes. On the 5th December, I will be very glad if you allow us to move into agriculture, because on the 5th December is a very special date for agriculture. So why not we learn together?
Okay, thank you so much. So inshallah, by God's grace, on 5th December, I will zoom into our backyard, inshallah. Okay, now the floor is open. If you have any question, you can unmute yourself, or you can type in the chat box. I'll read it loud. I think, let me enable. Okay, now you can unmute yourself if you wish to ask a question.
Hello?
Yes, you are.
Prof. I just want to ask a question.
Please, can you make yourself audible a bit? Hello?
Please come again. We can't hear you.
Hello.
Hello.
Yeah, hello. Yeah, Dr. Ibrahim.
Can you hear me? Hello.
Yes, we can hear you now.
Yes, I just want to ask in script. You talk about the headings and maybe the content of the table.
Hello.
Can you hear
Sorry,
me, sir?
I really cannot hear.
Yes. Maybe it's the network issue. Hello.
Yes, we can hear you now.
Okay. So with respect to the sources of the information contained on the tables, I can see some of the work after capturing the table in the manuscript or a paper. They normally write a source immediately under the tables. So I don't know how is that the importance of doing that? Is it necessary or otherwise, sir?
Okay, if I understood you correctly, you are referring to my talk, right?
Exactly, sir.
No, yes, I'm doing this to save myself from plagiarism.
Okay, sir. Okay, sir.
Because I'm telling you, these figures or tables or figures in particular are not mine.
Okay.
So this is just for the sake of presentation. But if it's your manuscript,
yes, sir.
if it's necessary, you can do. But if it's your own figure, then you don't need it.
Okay, so thank you very much.
You are most welcome.
Thank you very much.
Hello.
Okay.
Hello.
Yes, please, you are on.
Hello.
Yes, please speak.
Yeah, I want to really appreciate Prof for wonderful presentations from the beginning to the end. Please, my concern is for those participants that missed his previous lectures. How can we get the comprehensive dossier of the entire lectures from series number 1 up to this last series?
Okay.
I mean, at least, one way or the other, a participant may likely miss a session, whether session number 1 or number 2. So how do we have the comprehensive lectures from the series number 1 to up to this last one?
Okay.
Dr. Mutaka, I think you have all the series, am I right?
Yes, please.
So I think if you want to share with them, that will be fine.
Okay, perhaps
yes, I would like that.
For next email, inshallah, we'll add all the slides. And also, if you need the video on our Facebook, all the videos are there.
Okay, in the Facebook.
All right, thank you.
I mean the slides.
Okay, now please permit me to take the questions in the chat box.
Okay.
Okay. Please, may I know from Prof which unit presentation is right? Milligram per liter, or right mg per L? Prof, can see the check? Can click on the chat box?
Okay.
Yeah, in the chat box.
You mean the first one? Which one?
From I think Eric.
Okay.
Eric. Yeah.
Okay. I'm not sure how to address him, but Dr. Eric or whatever. What he's asking is whether p-value, all right? The p-value, the exact p-value of an estimate of 0.05 or 0.04, is it presented in table form? And then you say, can it be stated as less than 0.05 or whatever? I mean, okay, There is nothing wrong if you present your P level as less than 0.05, that is fine. The only thing is, if let's say example, if you have your P 0.045 and you say it's equal to this information, if we want to calculate and check your error, the one with the P is equal to 0.45 or P is equal to 0.54, we will be able to do so. But when you give us P is less than, because it's not exact value, we will not be able to do that. That is the only thing. But if you report and you say, can it be stated? Of course, the one that you have 0.045 can be stated as P is less than 0.05. But if you have 0.054, then we cannot say P is less than 0.05, because if you approximate 0.54 is just equal to 0.5. This is why it's better you use P is less than or equal to. So here you have to put a slash, then you can use 0.054. That means you are approximating it to the exact point. Now, if you look at the exact point of 0.054, now there is an argument here. If let's say you set out your research to be 0.05 and now we are having 0.054 and we approximate it to be 0.05, some will say it's right on the scale. So maybe they might reject and take the alternative. Some might say that it is right, it's on the scale, but we will accept. So that decision depends on you. But for me, as far as I'm concerned, the symbol that you are giving P is less than 0.05. That would not be appropriate to qualify 0.054. So rather, it must be P is less than or equal to 0.054. Then you can take that one in. So basically, it depends on the way you understand it. If I didn't make it clear, you can come back, then we can iron out one by one, then you can get it better. So Mr. Eric, is it clear to you? If not, come back again and we iron it out.
Okay.
Grateful. Thank you very much, Prof. It's all very clear.
Okay. The
What-
next one is Ali Nuhu. He is saying that, is it necessary to have a theoretical framework in a research? For me, I think it's necessary. It's necessary because if you sometimes, it depends on the nature of your work. If you don't have the framework, that means that you are not concept. You might be compelled to go into inductive reasoning or whatever, but a standard form of research, we might have a theoretical framework. If you don't have that, is fine. That is why it becomes inductive. By inductive here, you are opening yourself for what we call falsification. Unless you want to prove it to us that there is no any basis of the theory that you want to build on or hovering around, then it's okay, you may go ahead. But I think it will be difficult because it's going to take you that much of time for you to establish that kind of area. So that is, it depends. So it's going to be a long-term effect, that is fine. So that is the way I understand your question. If not, come back again and then we will reconcile the question. Now the next person is
sorry, Prof.
yeah.
Yeah, we take one from, we have two, they have raised their hand. Dr. Qasim. Qasim is there?
No.
Okay.
Yes, please. Yes, thank you for the opportunity. And let me also thank Prof so much for this information that he has shared with us from day one till now. Prof, I have a few questions. The first one is, I've seen, you see a reference if that is appropriate. My second question is regarding the use of.
Sorry. Can you repeat the first question because your voice is wobbling? I'm not hearing it. The first question again.
Okay. Sorry, Prof. It is raining here. Is it better now?
Yeah. Yes.
Okay, so the first question is, I think I've seen a couple of papers in which in the title, you see a reference. In the title itself, you see a reference. Is that appropriate? That's the first question.
Hold on. You mean title of the paper?
Yes, please.
In the title of the paper, not the title of
Yes,
the
please.
table or figure, eh?
The title of the paper itself.
Okay, I'm sorry, this is new to me. I've never come across this, so I don't think I can answer this question. It's very new to me. All right.
Okay, all right, thank you.
Right.
The second one is the use of p-value.
Yes.
Sometimes, or here you see in some of the thesis reports, you see p-value that is less than 0.00 or 0.01. So they just, instead of stating exactly what it is, they say p-value less than 0.01. And then p-value that is less than 0.05. So instead of stating what exactly it is, they indicate p-value less than 0.05. And so is it correct to use the two? Since your significance level perhaps was 95%, not 99. So if your p-value is even less than 0.01, is it correct to use that, or you can still say p-value less than 0.05? I don't know if you got me correctly.
I couldn't get you, but what I do know is, you know how to run a software, statistical software, right?
A bit of it, Prof. With you, I
Okay.
can say yes.
Okay, you correct me. I mean, what I do know is when you set your research and then you are going to run your data, some of the statistical software that I'm used to is SAS. SAS, the lowest that they start with will be 0.05. Now, when you run the data, you are basing on 0.05, but your research work, example, maybe you get 0.02. So in that case, you accept the alternative hypothesis, but your confidence level now is 98%. It's not 95%.
Exactly.
Now, when you set it up at alpha 0.01, and then you found out that your analysis it came out to be P 0.02. So here, because you set it at alpha 0.01, you were looking for a confidence level of 99. You have no choice now. You will have to reject alternative hypothesis and accept null hypothesis because there is no effect. So in that case, it is you who set it up. So you will have to decide now. So that is the way I understand it. But definitely you cannot report the two. So if I couldn't get you right now, let us filter out now. So am I in line with your question, or there is something that you want to reinforce? Then we can understand one another.
Yes, please. I will have to come again.
Okay.
So you have set your alpha level at p less than 0.05.
Yes.
So now you have found in your results, you have p is equal to 0.001.
Exactly.
Is it accurate to say p is less than 0.01 since your alpha level wasn't p less than 0.01?
Okay. Yes. So in this case, you don't have a choice. You have to stick to 0.05.
Okay.
You have to stick to that because that is what you run and you found out this. It just tells us that your confidence level is very high, but you peg yourself already. So in your paper, you should report alpha 0.05. And then you need to also understand this. This alpha level has been standardized in the whole world. We have three of them.
Yes,
0.05, 0.01, 0.0001. So whichever you take, whatever you get, you report based on that. That's it.
Okay. Thank you very much. Prof, my last question, you have mentioned it, but I just need a bit more
Sure, sure.
explanation or clarification on that. When you have a result that is, I mean, unexpected, and you are asked to discuss it because you have found something that is unexpected. Because it is unexpected, you usually will find little literature or scanty literature on that. So how do you handle such a situation, please?
Oh, yes. Now, assuming the result that you got is out of the ordinary and is credible, that's where we start from, right?
Yes, please.
Okay. I think these things normally they will happen. And if you have a scanty data or whatever, and it's something that is really new, I think the only way now here is for you to use some reasoning, deductive reasoning or whatever, and sometimes your own speculations a bit, but they must be convincing. You got what I'm saying?
Yes, please.
There will be some argument here, but if you are able to convince yourself and then you're able to convince people, and then it makes sense, even though it's not 100% established, that is fine because we can do detailed research sometime to come, then we understand it more. So I think that is acceptable. Hello?
Yes, Prof. I'm most grateful. I think I'm clear on that.
Can I give you a classical example to just see how it can contribute or can even set the tone or the debate? Let's go to the theory of evolution. Right. We have somebody called Jean Lamarck. Jean Lamarck was a German. And during the hot discussion about this evolution thing, even before coming of, what do you call it, Darwin. Now, they based, sorry, I'm sorry, there is a background here. So here, Jean Lamarck used something called giraffe. We all know what is a giraffe. Now, we do know that giraffe, generally, they are long-necked, very long-necked. But look at how Jean Lamarck started, because that time there was no information, so people are just reasoning. So Jean Lamarck was trying to explain why giraffes develop long neck. So his theory or argument was that sometime to come, the giraffe and whatever, they were getting their food from the ground. Now, as they start to run out of food on the ground, they have to get their source of food from trees. So what made their neck became longer was because they had to constantly stretch their neck, constantly. Those days they were short-necked. That means that they didn't have that kind of long neck. But because with time, they had to stretch their necks constantly to get food from the tree. That is how they became what? Long-necked. During his time, can you imagine that the colleagues or the academia applauded him and found that, oh, it makes some sense. But later, when Darwin came in, and when he came with the survival of the fittest concept, he tried to explain that, no, it's not like that. But it could be that during that time, you had short-necked giraffe and long-necked giraffe. You get it? So when the food got depleted on the ground, it was only the long-necked giraffe who were able to constantly stretch their neck and get food. So those who couldn't get food from the tree, they died. And this brought the concept of survival of the fittest. So what I'm trying to tell you now is, you see, Jean Lamarck used reasoning that was accepted at that time. But when we advance in knowledge, we realize that no, it doesn't work like that. I'm not saying you should believe in the theory for evolution, but I'm telling you the beauty of science. So what you have found, hello? What you have found could be very new and we have less information, but if you can give some reasonable information about it, that is fine, go ahead. Is it all right with you?
Yes, please, thank you very much, Prof.
Okay.
Hello, Dr. Muntaka. Hello, hello.
Happy. We have I think.
Dr. Munchaka, please let me talk in addition to what Prof just explained.
Okay, okay. Is it this Sulaiman?
Yes, yes.
Okay, all right. I saw your hand up. Okay.
Please, in relation to the explanation that Prof gave concerning this p-value. Hello?
Hello, please go
Yes,
ahead.
we can hear you, please.
Okay. I'm saying in relation to this issue of p-value, we are used to having p-value at 95% confidence, whereby we have either 0.05, 0.00, 0. Whatever. But I came across some empirical research work whereby you will see the values, to my eyes, they are insignificant. But the authors or the researchers keep reporting them as significant values. So my concern here is, are there certain exceptions whereby you will have some figures that are, for instance, 0.2, whatever, and yet is significant?
Okay.
What I mean, we are mostly used to this issue of 0.05 or less than 0.05 or something above, maybe 0.067 and the rest. But I came across some empirical research work that shows complete insignificant to my eyes, but yet they will be reporting them as significant values.
Okay. This is a very good point.
Is there certain exceptions, or are there certain softwares that, they find it normally bring out figures like that?
Okay. I think this is a very good question, Dr. Suleiman, and thank you for your contribution. What we will have to know,
yes, sir.
The paper that you read, example, let's say, assuming we are using 0.05, what was the P that they reported? Their P. Because you say it's insignificant, so I expect that their P value must be more than 0.05.
Yes, in fact, sometimes it is clearly shown 0. For instance, 0.2 something, but yet they will continue reporting as significant.
0.2, right? Let's say 0.2.
Yeah.
Okay. First, if it's 0.2 and they set their alpha level to 0.05, then there is a question mark. There is no any software that is a statistical analysis that will accept this. That's number one. But number two, what kind of analysis was done? I need to add on to this. What kind of analysis? What is a stepwise regression?
Yes, it is using multiple regression.
It's stepwise.
Yes.
Okay. If it is stepwise, we allow up to 0.15.
15.
Yes. If it's stepwise, not multiple. Multiple is different from stepwise regression.
Yes, apart from the stepwise, I even came across multiple regression that are using 0.2 and reporting it as significant.
No, that is not acceptable. It's not acceptable at all, because now their confidence is 80%. And if your confidence is 80%, it's not acceptable.
And yes, 80% level of confidence is not acceptable. So automatically, we should not have a significant result that is 0.2 and 2.3 or 2.4 or 2 whatever.
What the closest I know is stepwise regression allows up to 0.15, 15%. That one also has its own reason. You see, this is a problem. Did you check the credibility of the paper, the journal?
Yeah, actually, I can't remember precisely now, but I know it's a journal published in one of these European journals.
Okay, it can be European, it can be American, but sometimes the editorial board or the reviewers have no sound understanding of statistics. So,
except that.
Yes, so it could be like that. I have never seen this. I'm teaching statistics from first degree and to PhD level. I have never seen this, and it's not acceptable. You know why? If you use this to calculate your critical values, we standardize 0.05, 0.01 to avoid unnecessary argument. You get what I'm saying? So these are the critical values that have been accepted. Anything below this, you can accept. You may go ahead and accept, but actually, you know what it's telling us? you have a lot of statistical error in your work.
Yes.
Example, your replication.
That's exactly how I feel. That's exactly how I feel. In fact, I wasn't comfortable at all with those figures.
No. Such a person should. Okay.
Thank you very much.
Dr. Mutaka, if you may allow me to
thank you very much, Prof.
Add something to this present discussion, please.
A follow-up.
Just an observation. Hello,
Yeah, Prof, your hand is up.
Dr. Mutaka.
Yes, please.
Please, can you allow me to make observation on this one?
Okay.
Not my, this present discussion.
Okay, all right.
The way I understand his question, maybe he was not, or he is not clear. He was not able to differentiate between rejecting hypothesis using p-value or t-value. Some are using t-value, and usually t-value can be more than 0.1.
No, I understood very well. It's not t-value I'm talking about.
Okay, thank you.
I'm talking about p-values.
Okay,
t-value is different in regression. It's about the impact, impact.
I'm talking of the p-values, not the t, the p-value.
Just an observation. Thank you.
Oh yeah, it's a good observation, but I'm certain about what I'm saying.
Okay, thank you.
Okay, Prof, you are here? Okay. Madam Sylvia, okay, proceed.
Okay, Prof, I want to ask data analysis. When you are running your data from your charts, I realized that some of the data were far from the others. In that case, why, if it's me, do I have to maybe delete those data so that I can get exactly what I want?
Okay.
Or would include those abnormalities so that I explain them?
Okay. If you ask me, personally speaking, if you have an outlier, there are 3 schools of thought. One will say, if possible, repeat the experiment. That is one school of thought. And you agree with me that sometimes it might be too late or it might be too expensive for you to do that. Some might say that drop the outlier. Right. Now, if you drop, remember you lose some sort of degree of precision. That's one. Number 2, if you drop, the question is, will it affect your sample size or not? If it will not significantly affect your sample size, fine. Number 3 is, depending on your experimental design, sometimes you have no choice. You will not be able to drop it. Because sometimes, let's say example, you have 10 values. Let's say most of them are consistent. 1 or 2 or 3 are close. So if you drop one, you need to drop another one. You need to drop another one. You get it? So it becomes complicated. The third school of thought will tell you, depending on the type of data, you can do data transformation. And we have 3 types of data transformation. You have log transformation, arc-sign transformation, and angular transformation. So if your data qualifies, if that one will take me a lot of time, what is log transformation, arc-sign? So if your data qualifies for that, then you can do the transformation and you analyze your data. It will transform the data for you and bring it to the normal scale. But of course, you will lose certain element of precision. Now, if you try all of this and it doesn't work, and it qualifies for non-parametric, you can go for non-parametric if it qualifies. So these are the situations. So I can tell you yes, no, whatever. Because if, let's say, it's an outlier and you take the mean is not the true value anymore. Let's say one of the value is 1,000 and most of the range will be 100, 102, 103. If you add them together, your mean becomes higher. It does not mean the effect. So it does not represent your population. So you need to decide which one. I cannot tell you, but in my area, I can tell you if it's like this, you have to drop this or you have to report or you have to do whatever. So it's up to you. I'm not here to tell you drop or whatever, but the best thing will be cross-check. If you can repeat, that is the best.
Okay, Prof, thank you.
Yeah.
Okay, now we can move to the chat box questions, Prof.
Okay, what I'm seeing here is just comment. I'm not sure the last person, he said the role of theory in qualitative research may be minimal at the onset. However, the implications of the finding must be tied up to a theory at the discussion chapter. Fine. Then he said 95% confidence level is equal to 0.05, 99 is equal to 0.01. And you may choose to analyze using one tail or two tail. So I think this is a statement. I don't know what the, okay, that is Dr. Lee. Prof. Ali, you want, is it a statement or you are just?
Just a comment, sorry, just a comment.
Oh, okay. Oh,
Okay.
you are here. Thank you,
Yeah.
Check.
Prof, I have observation here, Prof, if you can highlight, little one.
Okay.
Prof, nowadays, some students use AMOS, SCM to analyze their data for survey, questionnaire, quantitative method, yeah? They don't use SPSS. When they use this SCM, they feel so pompous, thinking that they have done everything. Meanwhile, when you look at their theoretical framework, explication-wise, it's very, very weak. What can you say about this? Thank you very much. For me, I don't mind even if you use SPSS, it's okay for me. What is chunky? What can you comment on this? Thank you.
You see, this is a problem. What sometimes we do, that's a very good point. That's a very good point. Sometimes what we tend to forget is the robustness of the statistical software. These statistical software that we know them, you see, each will have, they all have their own strength, their strength and weaknesses. For example, I am very used to SAS, statistical analysis system, the one that is used in US, and most science-based kind of researchers, they use this kind of package. However, it's very weak in things like chi-square, not weak, it doesn't give you good kind of information. Now, if you go to SPSS, you will know very well that it's well grounded in your social science or whatever. So Dr. Ali, Dr. Ali.
I'm here, yes, Najinka, I'm here, yes. I'm listening.
Are you familiar with R statistics?
Say again?
R statistics.
R.
R.
Is it talking about the correlation or what? The relationship?
It's also a statistical software. Of course, it does correlation, whatever.
I'm more
and
familiar
also
with SPSS.
What I'm trying to say is you have to look at your work. Okay. And look at which software has its own merits and demerits. But for me, like my research I never use SPSS because it cannot answer my questions. Let's say if I want to do something like split plot design analysis, SPSS cannot handle that. So if I just go and key in my data and get the whatever, it doesn't work. So Dr. Ali, it depends on the nature of the work and the specialization of the software.
I agree, it's true. SPSS is mainly for social sciences. Yeah.
Thank you for the comment. And the other person also, I think is just a comment, I'm not very sure. I think is Prof. Ali the same. And then one is saying, please let me talk. Oh, is it you are the same person? 3.
Oh, no,
okay.
not
Dr. Suleiman was that.
Oh, okay. So I don't see anything. That's all that I can see from here. If you have any comment, please go ahead.
Thank you, Prof.
Because today is the last day.
The last day on this series.
Yes. Later we talk about patents and other things. How to get a research, international research grants.
All right, any comment? While waiting for further comments, please do write your names in the chat box for your certificate. If you have not done so, please do so. Some of your emails we received last they bounce back. So some of you did not receive the materials and the certificate. So please provide the correct email addresses.
Dr. Mutala, I think there are some questions. Okay.
I think somebody is asking about, I did mention of a stepwise regression. And this is the question, Prof. One of your responses, you mentioned the case of stepwise regression where a p-value of 0.15 could be used. Can you throw light on that? You see, stepwise regression is just like multiple regression. However, stepwise regression tells us, an example, let me give you this example. Let's say I'm selling a product, and then I want to see how advertising can help me to sell my product. So I decided to use internet, social media. Then I decided to use television. And then I'm using, let's say, radio, and then newspaper. So each of them, I'm going to invest in them to do my advertisement. However, I would like to know what will be the contribution of each medium of advertising on my sales. You get it? So here, if you go by ordinary multiple regression, it will just tell you maybe some are contributing to your sales, some may not, or whatever, but it doesn't tell you the percentage, the proportion. So if you want to know exactly how much will newspaper contribute, how much will whatever social media, television, newspaper, and then maybe internet. So here you subject it to stepwise regression. So long as it's quantitative, it will tell you, maybe it can tell you, look, I have all these variables and I have included them in your model, but you see, maybe internet kind of advertisement is giving you maybe 80% of the contribution. And then maybe, if you don't mind, radio is giving you just 2%. And then maybe television is giving you maybe 10%. Therefore, because you are going to decide whether you want to drop some of them and then focus on maybe the social media and then maybe television, then you drop the two, you need to be very, very careful with this decision. And 0.05 is too narrow for you to do this and to take the risk. This is why they expand it to 0.15. So within 0.15, whatever you take, the risk will be less. So here, in this case, let's say like I drop, let's say example, newspaper. Newspaper is contributing just 1%. So I can do without it and focus on the other ones. So this is where stepwise regression comes in. It's just like factor analysis, but stepwise regression is more quantitative, and then it must give you the model and the percentage. This is why we go more than 0.05, because if you go 0.05, some of the factors may contributing and you will drop them, and then you might incur some losses. So this is why we move to that level. It's not me, but that is how it is established. Thank you very much. I hope that is clear. And then the next person, I cannot see this, okay, I think that's all that I can see. The other ones, they are not questions actually. I don't see much questions anymore.
Okay. All right. I hope there are no further questions. It's already 10 p.m. Is there any last question we can take?
Somebody about the milligram.
Okay.
May I know from Prof which of these unit presentation is right? Milligram per liter or milligram per liter or milligram per liter? Oh, one is small letter. Are you talking about who is this? May I go back to this person? Dropenu, Eric.
Yes, please.
Are you around? Are you talking about milligram per liter small letter and milligram per liter big letter, capital letter? Is that what you want to know? Or is it a typographical error? Hello? Hello? Okay, if you are just focusing on this milligram per liter as a small letter, milligram per liter as a capital letter, actually what we want is you should use milligram per liter, the capital letter. That is the current unit. The small unit, that one is no more acceptable. So the current unit is milligram per liter, the capital letter. If you want to know about concentrations, we have milligram per liter, microgram per gram, milligram per kg, ppm. They all mean the same thing. I repeat again, milligram per liter, microgram per gram, milligram per kg, ppm. They all mean the same thing. But technically, if you are expressing your concentration in terms of weight, then you will have to use milligram per kg or microgram per gram. Then it becomes content. But when you use ppm and you use milligram per liter, that is what we call concentration because it's based on volume. So I'm giving you extra information. If in case that is what you want, yes. But if you are just concerned about small letter L and capital letter L, the current is milligram per liter is considered as the current, and that is what should be written in your manuscripts. Thank you very much.
Okay, any final question or thought?
But you may also want to ask.
Okay, Dr. Sulaiman. Okay.
Actually, it's not addition to anything, but how do we get the contact of the prof anytime we would like to discuss issues with him?
My email is there, but if you want my phone number, Dr. I don't mind, Dr. Mutaka, you may share it with them.
Okay.
Okay. Thank you very much.
I think WhatsApp will be better.
Please, what is the name in your Facebook page?
Unfortunately, I'm not active on Facebook.
No, not you. I mean the Dr. Muntaka, the Ghanaian
Association of Ghanaian Students in Malaysia.
Oh, in Malaysia, that's the name.
Yeah, Association of Ghanaian.
Because you said that I'm the very person that requested about the comprehensive videos and the slides, and you say it's available in your page.
Yes.
Okay, one can easily download them there.
Yes.
Okay, thank you very much.
Okay.
In fact, we really appreciate the contribution of Prof to knowledge.
Okay.
That is so wonderful, and we are really grateful.
You are most welcome. Thank you all also for coming, and we all share. Like I keep saying, I think it's not that I know better. I don't know. We just have to discuss. Some of you are better, just that you are trying to be humble. So in the spirit of academia, we will have the room to share, and nobody is above nobody. So thank you all, and then we will meet another time.
Thank you.
Hello.
Yeah, Prof.
Yes,
Yeah.
I know this issue has to do with grant.
It's a passionate request.
In the village.
Hello.
Prof, there is a passionate request.
Okay.
Whether your face can be seen.
To be very honest with you, somehow I don't know why in my lectures until today I don't show my face. I don't know. Somehow I'm not comfortable with my displaying.
All right.
Yeah. But for the sake of today, maybe just 1 minute. I seldom do this.
Oh, okay. I want to know you. Mashallah.
Okay, so this is
Prof, if one need to contact,
wonderful.
Prof, if one need to contact you, how can we get you, sir?
My email address will be with Dr. Mutaka, and then my phone number is also with Dr. Mutaka, and Professor Ali is here. He's my senior brother here. If you want further details, we can also get from him. He's and then I can see we have also Brother King Abbas is also here. So thank you all of you for coming. And then we'll meet another time, inshallah.
Dr. Mutaka, please, I would like to have his email and the phone number, please.
Okay, the email is on the slide. Then the phone number later will provide. Okay.
Okay.
So, Dr. Muntaka.
Yes, please.
Please, I really appreciate your contribution as a person and also the Ghanaian Student Association. I would like, Prof has talked about the issue of international grant.
Okay.
Yes, I know by the time you people get something like that, it will give us more opportunity to discuss on so many research issues. Hello?
Okay, yeah, we're hearing, Doctor.
Yeah, I'm just appreciating you people and also telling you that we wish by the time you get more grants, we can also benefit from it by having lectures.
Okay, all right. Thank you so much.
Yes, thank you very much.
Yeah.
Okay.
Like I did say, I don't mind if you want us to talk about international grants, if you want us to talk about patents. I mean, a whole lot of things that we could be talking about. I don't mind at all.
Okay.
It's Prof. Keep sharing, Prof.
Yes, thank you. Brother Abdurraouf, I see you here. Muna Geswa, I hope you are fine wherever you are. Thank you very much. Do you permit me to leave now? Because we are ahead of...
You are free to leave, Doctor.
Okay, thank you. Assalamu alaikum.
Wa alaikum assalam.
And then good day to everyone.
Okay. The topics you need for next session, perhaps you can forward. Okay. You can forward the topics you are most interested in for subsequent sessions. Okay.
Mr. Mutaka.
Yeah.
I sent the correct email, but I didn't receive the certificate, so I don't know why.
Hey,
Dr. Mutaka.
Yeah. Oh, your email is there, your certificate is there. I know, I remember. Okay, we'll check again. Okay. All right. Okay.
Hello. Hello,
yes.
Good afternoon, Dr. Mutaka.
Yes, please.
Yeah, Dr. Mutaka. Dr. from Nigeria.
Okay.
Mutaka, can you hear me?
Hello.
Please allow Dr. Lukman to finish.
Yeah,
Okay.
from Nigeria, Zaria, precisely in Kaduna State.
Okay.
I really appreciate the lectures. It's a job well done.
Okay.
I lecture with the Gombe State University in Gombe State, but I reside in area.
Okay.
I must appreciate your effort. It's a good one. Please keep it up. May Allah reward you abundantly.
I mean, thank you so much. Okay. Okay. Bravo, Baka. The floor is yours.
Hey, Dr. Mutaka, we have requested Prof to show his face, and we have not seen your face. We will appreciate if you can bring yourself to us.
Okay, let me keep it in suspense. Next session.
Thank you so much, but I have been raising her. I don't know, you have not seen me. Maybe I will send the email of the Prof will be shared to us. Maybe I can use his email to send my question.
Yeah,
thank you so much.
On all the presentation slides, we have his email there. Yeah.
Okay, so I will use his email. I can maybe see how I can reach him through the email. Thank you so much. Thank you. We appreciate.
You are free to contact. He's very simple, willing to help and share. All right.
That's nice.
Next session, you.
We have not seen you.
Let me.
Okay, so I will see
you next session.
Okay, so thank you so much. Thank you so much. Thank you so much.
All right.
Bye.